Thank you so much. And thank you to those of you who've joined us today. I'd like to welcome you to the MSG ERC virtual collaborative. The topic today is novel antifungal therapies and stewardship. This is a program that was carved out of our biennial program as a standalone webinar, which we're conducting today. I want to again thank everybody for being here, and especially thanks speakers for their willingness to change and present their topics today. We have a really interesting set of speakers and topics, and really looking forward to all the information that's going to be provided and exchanged. Now, there's not a lot of housekeeping issues here, although to the speakers, you really must remain on time. We have a tight schedule, as you always can imagine on these types of webinars, and so I will expect you to be done at the end of your time. Most of you have 20-minute slots, and we'll have time for questions and answers at the end of each talk. So that's how that will work. And then for individuals who have questions, you can either use the chat or raise your hand, and we'll acknowledge you, and we'll get to every question that we possibly can. Now, we'll have a series of presentations, prepared presentations, but then to really kind of wrap this up today, what we're going to do is something that we haven't done before, and that is have breakout sessions. And these breakout sessions will be really determined by compound. And I believe that one can go from one breakout room to another, I think, but Debbie can clarify that for me. But basically, these will be determined by a new compound, and it'll be co-monitored by a representative from the company, from the pharmaceutical company, and one of the members of the MSG ERC. So I think that's really going to be just kind of an open session to learn about drug X, whatever really you fancy. And then we should be done in 3.5 hours. Okay. Debbie, was I muted during that, or did anybody hear me?
You cut out a bit. If you just want to go.
I don't know what. Okay. I don't want to repeat all that again, but I will keep people on time. Questions and answers. Raise your hand or chat, and then at the end, again, we'll have really an open forum where you can break out into separate rooms and be really just in a conversation with a representative from a pharmaceutical company that's representing a new agent, together monitored by one of the members of the MSG ERC. So with that, I'm going to introduce the first speaker, and our first speaker today is Maurizio Del Poeta. And Maurizio is Stony Brook University on Long Island. He's going to talk about early antifungal development from target to startup. This is an area of his passion, and there's really no one better to talk about this than Maurizio. Thank you so much for being with us today, Maurizio.
You're welcome. You see my screen?
Yep, looks good.
Wonderful. I do have an interest at this time. I really hope it will become financial soon. I am affiliated with Microread Technology and also Novartis. I graduated in medicine in '92. I started the residency and I did fellowship in infection diseases at the University of Ancona. And I did some time in San Antonio to start the in vitro susceptibility testing in the fungal testing lab. I did an ID fellowship at Duke University with John Perfect. And that's when I was excited about doing research and start antifungal there. And John hired me for a research associate for 3-4 years. Then I met a lady there and I got married. So she moved to South Carolina. I moved to South Carolina and I went through the professorship there. And then I moved to Stony Brook because my wife moved to Stony Brook, so I had to follow her. And a few years ago, we co-founded the microread technology. And I will tell you a little more about this. I study mechanism of fungal pathogenicity and particularly on lipid pathway, on sphingolipid and glycolipids. And we also study host susceptibility to fungal infection, particularly on sphingomyelin synthase, sphingonicanase 1, sphingosine-1 phosphate. But the talk today is about antifungal development. And we have 3 projects. One is going after the vesicular trafficking, and this is going after fungal glucosylceramide, the sphingosine synthesis. We have another, this is the most advanced program. We have 2 additional programs, which is targeting specifically CEL1 and SGL1, which are 2 fungal specific enzymes, but we are not talking about this program. Although the company, microread technology, is involved in all these antifungal programs. This is a collaboration between microread technology, the Center of Biotechnology, my department, microbiology. We are fortunate here at Stony Brook to have an Institute of Chemical Biology, Drug Discovery for MedChem. And the collaborative effort is with the FDL in San Antonio and the University of Cincinnati for some PK study we do there. And with Fiocruz, because Fiocruz is interested in veterinary use of our compound. A few years ago, we discovered a new class of compound called acylhydrazone that has this peculiar phenotype. It decreases the synthesis of a certain type of sphingolipid called glucosylceramide that you see here on a TLC. And a TLC is a thin layer chromatography, it's a pulse-chase labeling in which you are able to visualize a certain type of lipid. In this particular type of TLC, you're visualizing sphingolipid. And you can see that there is a decrease of certain type of sphingolipids and an increase of certain type of sphingolipid called sphingosine. What happened is that this type of sphingolipid, particularly sphingosine, is extremely toxic to fungal cell. And therefore, when you are increasing these sphingolipids, the cell are dying. And this was demonstrated by a pulse chase labeling and also steady state labeling using mass spectrometry. And so we started a big program and that are looking at combinatorial strategy. These are small molecules. And we look at the different synthesis and different possibility to create a much better molecule than the one that we are screening from the synthetic library, which were done using the Cambridge library. And at the end of the SARA screening, at the end of the combinatorial chemistry, we screen about 400, 450 compounds. And here is the flow screening that we use, minimal inhibitory concentration, the killing activity, the toxicity in mammalian cells. And I would like to point out that we were extremely stringent with our assay, looking at a selectivity index greater than 500. These are small molecules which might be toxic, so you really want a very big therapeutic window. Anyhow, at the end of this, we end up to have a 5 representative compound that has a very, very good activity in vitro, in vivo. They have a very good PK and not much toxicity at least in animals and mammalian cells. And so when we had these, there's a question of whether or not you want to start a company and how do you go from a target, from either your product or even from your idea to a startup. And one possibility that you can do is just to go online and just type from idea to startup. You can see that there is many possibilities there to learn a little more. What does it take? And if you try to read the certain types of document, you will see that there is a different type of startup and an LLC or limited liability company doing business as or a corporation, and whether or not you're going to go with an S corporation, with a C corporation. I will be happy to elaborate this later. But anyhow, online, I will give you a lot of information but also a lot of confusion. You may have a 36, 35 steps to form a startup. Some other will tell you that you can do it in 13 steps. Some other will tell you in 12 steps, and some other will tell you in 9 easy steps. And I can tell you that if there is anything about these things, that they're not easy. But I decided to go and talk to two charming faculty that in my department, because they had a startup. So one was Eckhart Wimmer, and the other one was Sumita Baduri McIntosh, and they had Codagenic and Ocus Locus. You can go online and see what they do. But they told me, you know, Maurizio, I think the first thing you do is you should go to talk to the New York Center for Biotechnology. And I said, wow, that's nice. I said, where is the Center of Biotechnology? Do I have to go to Albany or to New York City? And they said, no, it's just behind your building. So what it turned out is that the Center of Biotechnology for the entire state of New York is located on Stony Brook campus, and it's really behind my building. And so they have a Long Island Science Incubator right here on campus. And I learned that this center already helped over 40 companies during the last 15 years to start up here from Stony Brook University. And they already put several drugs in the market, and particularly a lot of devices for orthodontics. So, and they also have a program of bioentrepreneur in residency, in which there are people that are helping the faculty to start up a company. And so I decided to give her a call, and I met with Lee Leo, which is now a director of business development in a company. He's not working here at Stony Brook anymore, but I met with him and I told him who I was, and I told him what we were thinking to do. And he gave me a paper to read. And he said, it was really nice to meet you last week. Please find attached the paper that discussed about faculty involvement in entrepreneurship. And happy to discuss further if you have any questions. So I immediately, obviously, read this paper. And this paper was about how much risk are you prepared to take? And it's a Nature paper published by Van Dyke. And he's talking about any faculty member wishing to commercialize the discovery of technology should contemplate the pros and cons of pursuing an entrepreneurship route or license it to an outside entity. And basically, the paper is talking about the 5 P's that you must have. These are the people, the patent, the papers, the preliminary data, and the funding. Believe it or not, this is actually the importance of how you should start thinking. The funding is not the first important thing. You know, people think that the money are the first important, but it's actually not. Because if you have the people, if you have a patent, if you have a paper, the preliminary data, the funding eventually will come. The people is absolutely so essential. The meeting, the stature that you have to develop this startup is absolutely essential. If you don't have a patent, there is no way you cannot sell something that you don't have, you don't own. The paper is more about what kind of stature you have in the field, more than a specific paper about the product. That is in the preliminary data, which obviously is to be strong. If you want to sell something and you want to propose something, it is to be definitely better than something that is already out there. And if you have this one, you know, the funding eventually will come. And so I read the paper, I decided to give it a call to Lee again, and they had a meeting with me to talk about more about the startup. And at the meeting, I asked a question. I said, you know, it's funny that there is no disease. And they look at each other and they look at me. And I remember one guy said to another guy, I said, yeah, this guy needs some business training. And then they left the room. And so I was left there with Lee, and he told me, don't worry, Maurizio, we're gonna talk about this. But apparently, in some cases, I mean, in many cases, disease is not needed. I will be happy to elaborate this a little later if you want to. But obviously, this is not true for the other drug that we have. But and so what we decided to do is a key important step that they asked me to do is to definitely decide my role in this startup. So you can have a role in which you can be just a consultant, but in my case, I really wanted to be a little more involved in the formation of a startup. So I really wanted to be a guy that produced the data, the science, because that's what I do. And so I decided to be a chief scientific officer. And then they asked me a question whether I wanted to be a founder or or not a founder. And there is a difference between that, because if you want to be a co-founder, then you own the share of a company. Whereas if you don't want to be a founder, then you don't need to be the share of a company. But if you want to be more in control of a company, a co-founder is definitely a way to go. The last thing that you want is to have somebody coming, get your product and sell it to someone else. So we decided to select a CEO, and to select the CEO, they asked me to first define very, very clearly the goal that I wanted to have, and really to write down a strategic plan, which is something that we did. And so together with the Lilio and together with the Office of Technology and Industry Relations at Stony Brook University, we decided to interview the bi-entrepreneur in residence that was here at the Center of Biotechnology. And we selected Brian McCarty because Brian McCarty, and we interviewed three or four people. We selected Brian McCarty because Brian McCarty was coming with John Malamo. And John Malamo has 30 years of experience in industry relationship, and he was the vice president for drug discovery at Cephalon, who then was acquired by Teva Pharmaceutical, and he's still working for Teva Pharmaceutical as a consultant. And so John Malamo has a very large experience in bringing a drug into the market. He brought about 15, 20 drugs into the market. So that was very good for us to have John on board. And so the next thing that we wanted to do is to have a stockholder agreement. And this is important for a foundation of a startup company. If you want to be a co-founder, you really need to be in a position in which you own some share of the company. And this is very important because if you don't have this, then the CEO or who owns the share can sell the company anytime you want, anytime they want. And this stockholder agreement also is something that needs to be evaluated by a lawyer. The lawyer needs to look at this to see whether or not it makes sense to have this type of agreement. Other agreement that needs to be in place, something that I have to become more familiar, was a collaborative agreement between Microread and SBU or any other entity, because I am a faculty, and as a faculty, I need to disclose as a state employee everything I do for the company. So there is a management plan between me and the university that allowed me to do industry work. I cannot work, for instance, more than 20% of my time for Microread. I need to become familiar with what means a material transfer agreement, with the confidential disclosure agreement, non-disclosure agreement, confidential agreement, dual data use agreement, data transfer agreement, subcontract agreement, sponsor research agreement, collaborative agreement, consortium agreement, consulting agreement. So you really need to be familiar with a lot of agreements about this issue. Conflict of interest and the management plan is in place between me, Stony Brook, and Microread, so that I can do whatever I have to do. Clearly, there is a lot of keeping record of everything that needs to be done between a faculty. Again, this is particularly in my case because I am a co-founder and the chief scientific officer. If you want just to be a consultant, you don't need to do all of these things. The IP, a provisional disclosure of a patent, is absolutely essential because there is nothing that you can say or you can disclose without prior disclosing this to your patent office at your institution. It's a big mistake to disclose anything that you do with the company before disclosing to your patent office. And so I have to become familiar with the key topics that are different than normal, particularly on drug. development, and particularly on grant development. Instead of background and significance for grant, for instance, there is what is the need for your product, what is your proposed solution, why this product is different than the current standard of care, what are the team, what is your project plan, what is the commercialization plan. Even if it's early, you need to be able to understand the value, the expected outcome and income. I remember that I gave a talk at the Life Science Summit here in New York, and when I was telling that the drug was killing the fungus in 3 hours, the guy stand up and said, "This is not good. This is not good because it's killing too fast. How long do you think you can get the therapy? If a therapy is only for 1 or 2 days, we don't make enough money." So this kind of question, you may have this kind of question. You need to be prepared how to answer this kind of question. A too good drug might be not business potential. You really need to have some IP protection again, production and marketing plan, revenue stream is very important if this is a business. Transition plan and regulatory path, potential risks and mitigation, this is similar to other type of grant, timeline, milestone, funding requirements, budget justification. Has to be very detailed because this type of grant for commercial, for business grant are normally require a detailed budget and not a modular budget, such as when you're applying for a classical R01 or something. For microread technologies, currently on the preclinical development of the acyl hydrazone, and we hope that during the next 12 months, we are finish up our preclinical portfolio to be submitted for the FDA for IND. And we are working very heavy now on the target product profile, particularly on the type of formulation. We have an IV formulation, we have an oral formulation. We are developing now a topical formulation. And we are engaging with the strategic partner for development based upon a value proposition, which is currently superior to the standard of care. And this is where we are at this time. So I would say pros and cons to start a startup. If you really go and analyze the pros and cons, the cons are really a lot. There are many, and I would say you're not going to do it. I would say, you know, do you really have a passion and excitement to do this? This is something totally different. This is something personal also, and it gives you opportunity to learn to something different. It makes you also a little more creative. You are more independent. You will add a lot of responsibility. Talk to your wife or your husband first. But I think that you will be able also to do a lot of different things that, you know, you hang out with a lot of people that will have a lot of more money than you. That's for sure. And I will suggest to go and talk to any other people that has a startup. There is a state center for biotechnology in every state. So that's something that you can reach out for suggestion. I would say, you know, a basic understanding on IP, COI, and agreement is absolutely a must before doing anything. And definitely you need to be prepared to do a lot of fundraising, because to advance your idea, to advance this type of idea, you really need to convince a lot of people that it's really worth to spend a lot of money. So fundraising is really, really important. And so I will conclude by remembering Mike Rinaldi, because I started this antifungal development in his lab, and I just want to say that ciao paisano. This is my group, Stony Brook, and we have several people working on mechanism of action, formulation, and development. Iwa Ojima here is the director of the Institute of Chemical Biology. Nathan is helping at FDL for some study, and Melanie and Pankaj at the University of Cincinnati for PK. We have this collaboration with the University of Rio de Janeiro and Fiocruz, again, for veterinary use of our drug. And obviously, I would like to thank Mike Reed, Brian, and John for their support throughout the years. This is something that I started a few years ago and is going very well so far. And the funding that come from the REACH program here and the NIH R1, R41, the CTR, and the DoD funding for this antifungal development, which is very generous. And I also would like to thank for this, for you guys participating in this, and I would like to thank again Peter for inviting me.
Thank you so much. Thank you so much, Mauricio. Very, very interesting perspective. We don't, get that a lot from our investigators. And so we'll open this up for discussion. We have about 7 or 8 minutes for questions or comments. I'd like to ask 2 questions just to kind of get us started. Number one, when one starts up a company like this, and especially when you're talking about, small markets and so forth, is the ultimate goal to get bought up by another larger entity, a deep pockets entity? I mean, is that really where the investment pays off? Is that the ultimate goal of those who develop these small startups?
Yes. So, for the state of New York is fantastic. We have Alexandria Center here at New York City. It's the place where we are exposed as a small company to big company. And once a year, we have this meeting with them and say, which of these products are they willing to go forward. And you are definitely absorbed by them. Either you are selling to them, you may find enough support to say, hey, I have a $50 million, $70 million investor that is coming in to do a clinical trial with you. Yeah, then you can do it. But if it doesn't, look what happened with Amplix, for instance. Merck eventually, Pfizer eventually came in. And in my opinion, it's very extremely difficult that we will be able to commercialize this compound. Eventually, we will have to sell to a bigger company, which is fine.
Yep. Okay. And the other thing that came to mind, you mentioned that one of your compounds, or one of the compounds you were looking at, you suggested that it worked too well, killed fungus too fast, and that it limited its commercial value because it wasn't going to be given for multiple days. Were you being tongue in cheek, or was that a real consideration?
No, it's a real consideration. Yeah.
Interesting. I think most people in the pharmaceutical industry would just adjust the price to accommodate that. But anyway, let's see if there are any questions in the chat. There are a couple of comments.
Yeah, I see that there is a comment about NIAID. NIAID was extremely supportive. But for instance, the study that we have done with San Antonio was through NIAID. NIAID has some subcontract sometimes. So if you have a compound that you want to be tested, and you can reach out to a program officer, and you have to submit a proposal, and if approved, the program officer can subcontract an entity. In our case, was FTL for this testing for us. I know that Dana is connected right now, so maybe she can elaborate a little more. But that's something that we have done with several of our compounds throughout the years. And so, I think, yes.
Hi, Maurizio. I think Erin Zaytuni, our preclinical services manager, is online, and we could upload something, or we don't want to take up much time, but we do encourage folks to reach out to us. These are in-kind services, free services, and it doesn't take a year to review to find an answer whether you're going to get support or not.
Yes, absolutely. So we have used this service a couple of times.
John Rex also mentioned that there's the California Life Science Institute that's also an accelerator for support and development. There's a question from Abyssine. how do you gauge and set up a safety net from any legal liability when considering being a co-founder versus solely a consultancy role? So,
Yes.
you know, a matter of ultimately responsibility, I suppose, is really kind of what it boils down to.
Yes, so it's a key to talk to your patent office and industry's relationship. You need to know exactly what, and this is different from university to university. In my case, for instance, the state university, I am bound to do certain types of plan and certain types of things that other people in another university may not be able to do so. So it is something that your office is going to tell you what you can and what you can do. So it's absolutely essential. And this has to be done before doing any type of agreement with your outside entity. Otherwise, your university may say, you cannot be a co-founder. You just cannot. You cannot hold any share. And if they won't allow that, there is nothing you can do. It's a breach of your agreement with the university. I like it because in the state of New York, this is possible. And because of the Center of Biotechnologies here, maybe it's possible. I don't know about that. But you know, when I ask this question about whether or not I can be a stockholder, they said yes. And this came out with the lawyer, because the lawyer said, Maurizio, why you want to give away everything? You know, some type of this yeah. That was really funny, by the way. When I contacted the lawyer told me, the first 30 minutes is free. The next hour is $1,200 an hour. I said, you gotta be kidding me. There's no way I'm gonna pay that. Don't worry, we're gonna solve this in 20 minutes. Okay, then. So, but the point is that you know, it's really essential that you have a say whether or not about what you have done, because otherwise you're gonna give it away. And a CEO may come in and say, I sell it. And there's nothing you can say.
Maurizio, I want to thank you again. And I think we'll move on to our next speaker. There are a couple of comments that are worth looking at in the chat box. And I would encourage you to pursue some of these references. A second talk actually will be from John Rex, a good friend, an old friend. And John is currently serving as Chief Medical Officer for F2G, another relatively new startup biotech firm. And John's going to talk about the regulatory landscape. He has a lot of experience with dealing with, you know, NDAs, getting drugs through both EMEA and also the FDA. And so he's going to give us his unique perspective on the regulatory landscape of antifungal drug development. Thank you, John, for being here.
Thanks, Pete. And am I on? Can you hear me?
Yep, we can see your screen and you sound great.
Wonderful, thank you. So I have given these slides to the group to share, so everybody should get a copy of this. Let me just start with say thanks for asking me to comment on this. So here are my coordinates, and you don't have to drop me a note because you're going to get the slides. And this deck is a mashup of things I've learned over the past 40 years. I have been poking at the question of developing new antimicrobials since I was a fellow with Jack Bennett. And over time, I've gotten to try it from both the academic and the pharmaceutical regulated industry side. And Maurizio, I really enjoyed your story. That's exactly right. And I will say that venture capital, you start off with people. In venture capital, we often say we bet more on the jockey than we do on the horse. Because there will always be problems, and what we want to know is that the team has the insight to figure out what the heck to do about the problems when they pop up, because they always pop up. So this story is going to come out of all this. Series of learning objectives, and let's just jump right in. So learning objective number one is to understand the rules for drug approval and why antimicrobials are usually developed with non-inferiority design trials. And the foreshadowing here is that infections are lethal, and we must design to avoid superiority. So if you look at the big two, FDA and EMA, I'm really going to focus there. China and Japan chart a very different course, but most of the rest of the world follows FDA or EMA. FDA has a traditional approval pathway and a thing called accelerated approval. Is really very, they're built off the same thing. Both of them require substantial evidence of efficacy based on adequate and well-controlled data. A traditional approval, the efficacy is based on an endpoint that a human being can perceive directly. Feels, functions, survives is the mantra. Accelerated approval is based on a surrogate marker that predicts how you will feel some years in the future. And the classic best example of that is the HIV viral load test, which has been a standard tool for predicting what will happen in 20 years, but you don't wait 20 years. So you approve the drug based on the marker. And then you do a phase 4 confirmatory trial. I'm going to warn you that this sounds on the surface a lot like EMA's conditional approval, but it's got there's a little different twist. So it's important to just keep the language straight in your head. Accelerated approval is about a surrogate. We're also going to talk about an idea called LPAD in just a moment that's distinct to the FDA. The EMA has a different basis for its approval. So on the previous slide, just put it back up, make you just read it. Substantial evidence of efficacy based on adequate and well-controlled data. Adequate and well-controlled data. Repeat after me. EMA, however, it says it's about the balance between benefits and risks of medicine, and that's the key guiding principle. And that it actually gives EMA a little bit more flexibility to decide what is enough. EMA has two other kinds of authorizations. One's called conditional marketing authorization, where the approval is based on limited initial data, and additional data are possible and are required. You will have to go and generate them. And it converts to a standard authorization when you provide those comprehensive data. So it's not quite the same as accelerated. And then there's marketing authorization under exceptional circumstances, where the approval is based on limited initial data and additional data are not possible or expected. Now let's come back to the FDA and talk about this thing called LPAD, or it really ought to be LPAD because it's the limited population antibacterial and antifungal drug pathway. It's important that both of them are in there. And this is a deal where you've got a drug for a serious or life-threatening infection that can be used in a limited population with an unmet need. And it permits FDA to approve if, and now this is going to start to sound a little bit like a mashup of EMA with FDA, if there is a positive benefit risk balance in the limited population, even though insufficient data exists to conclude that there's a favorable benefit risk profile in a broader population. FDA will take into account the severity, rarity, or prevalence of the infection that the drug is intended to treat. And this all becomes very important for fungal infections because they are uncommon. It takes a long time, as we'll get into in a second. As an aside on LPAD, it's not an easier pathway. It's not a way to fix a failed program. As for any approval with the FDA, you must show substantial evidence of effectiveness and sufficient information to show safe use as labeled. There are two compounds that have been approved to date using LPAD. One is an inhaled product for Mycobacterium avium intracellular pulmonary disease, and the other is a compound for XDR-TB. Now, talking about data, these three slides, where do you get your data? So here we're going to do a little sidebar into some core ideas you need to be familiar with. Trial Design 101. There are only two study designs in the entire universe. There is superiority and there's non-inferiority. Superiority is the easier one to understand. X versus Y. X is going to beat Y. Test versus placebo. Test versus standard of care. It's preferred design. The result is unambiguous. The trial itself is self-validating. X beat Y in the population of patients that I enrolled. Non-inferiority, on the other hand, is messy. X is about the same as Y, and X is not too much different from Y. It's got this ugly title, non-inferiority. So everybody likes the idea of better and superiority. Nobody likes the language of non-inferiority, but almost always in antimicrobials, we're going to use non-inferiority because of the paradox of antibiotics. While we want new drugs for bad bugs, and the superiority of new is easily shown in the lab on the basis of MIC testing or in animal models of infection. You start asking for clinical data, and you have a problem. Trials must usually be designed to avoid superiority. We're going to have to use this non-inferiority primary hypothesis because, just think for a minute, you pretend you've got a limb-threatening infection due to MRSA. Would you be happy if I came to you and said, I'd like to randomize you to methicillin versus my new drug, and we need some people to be on the methicillin arm so we could show that the new drug is better than methicillin. You'd say, you are mad. I want linezolid or vancomycin. Thank you very much. I want to keep my leg. And indeed, when you're doing a trial, you have to avoid enrolling if you think either one of the arms is no good for your patient. Now, this why not superiority? I spent hours and hours talking to people about this, and you can do it in some settings, like migraine. You can do a delayed effective therapy study. Your head hurts a little longer, but it didn't hurt you. Placebo for cancer, we don't do that, but there's usually room to improve on 5 or 10 year survival. The thing with infections is we cure them, and once you're cured, it's not possible to be better than cured. You're done. But still, this idea of non-inferiority is confusing. I want a better drug. I want to do studies on resistant bacteria or resistant finding. I understand your intuitive wish, but nobody wants to receive predictably ineffective therapy. Just keep this in mind. And in fact, the non-inferiority studies are very powerful. You say, how does the new drug work in an MDR infection? Well, unpack it. Does new work when the pathogen is resistant to other drugs and susceptible to new? Well, if you do a properly designed modern non-inferiority study of new versus old, you convincingly show that new works as well as old when old is still active. And if resistance to old doesn't matter when you're treating with new, which it shouldn't, it's susceptibility to new that matters. You know that new will work when the pathogen is new susceptible. It doesn't matter about the old drug. So by being able to do non-inferiority studies, we stay ahead of the game. So that's the background that leads to this as sort of the general pattern for new traditional antimicrobials. You do one non-inferiority RCT versus a good comparator in a setting where you can do something, in case of bacteria, complicated UTI, for example, and you've got a lot of supportive evidence. And then you do a parallel study in highly resistant pathogens. And here you do what you can. You randomize versus best available therapy, if there is a best available therapy. Otherwise, maybe it's just a single arm study, or maybe it's it's so small you can't even do any kind of randomization. Examples here include plazomicin and meropenem vaborbactam, both of which did a complicated UTI study, and then they did a highly resistant pathogen study in parallel. The approval tends to be based on the non-inferiority RCT. LPAD might not be used at all, and was not. And the data from the highly R study does not routinely appear in the FDA label, though hints of it may appear in the EMA label. Okay, last of the ideas, endpoints. This is a pop quiz. Pick the valid endpoints for drug approval. All-cause mortality, improvement of signs and symptoms of pneumonia, drop in HIV viral load, conversion of blood cultures to negative, invasive aspergillosis, fall of serum galactomannan from greater than 1 to 0. Tick tock, what do you think? Here we go. The answer is the first 3. And the reason is that an endpoint must be a reliable and reproducible measure of how a patient feels, functions, or survives, or it must be a surrogate for that. The first 2 are classic clinical endpoints. All-cause mortality is easy to measure. And while you have to do some work to develop rules for symptom-based endpoints, you can do that. The third one I've already mentioned. This is a well-defined surrogate endpoint that was used for many years as the basis for accelerated approval in the US. And there's a great read in the links below. It's actually, with new HIV drugs, you go straight to standard approval now because HIV viral load is now known to be so good that we don't need to fool with accelerated approval anymore. The other 2 fail because they're inconsistently linked to the disease state and progression. Yeah, blood cultures do need to go negative, and yes, galactomannan does need to go down, but it isn't 1-to-1 in the way the other things are. So we don't use them. Okay, so what are the time, cost, and feasibility implications of different kinds of clinical data, and why is it that non-inferiority trials cost so darn much? Preface: it can cost a whole study estimate can be $100,000 to $200,000 per patient. So it's because non-inferiority studies tend to need a pretty good size sample, 3 to 350 per arm for standard parameters, 20% failure, 90% power, 10% margin. I know superiority designs are smaller. Yes, I know that, but you can't do them, at least not ethically. And the key cost driver is the number of sites times the duration of the study. And unfortunately, you have to have a lot of sites because infections move quickly, and you can't usually transfer patients to a master site. There's not enough time to get them there, and that other hospital doesn't really want your resistant pathogen patient. Open sites are costly. Most sites contribute little. The graph on the right is from a recent paper by Johan Meritens and IA study that demonstrated that most of the sites contributed very few patients. But it's hard to predict performance, and you do want global coverage, so you kind of got to just have that range, and that's just the deal. But time is costly, project management costs, and actually a curious one, providing and maintaining local supplies of unexpired drug. I got to keep in this study, they had to keep 91 sites supplied with current fresh compound. Really an interesting problem. So you take the whole study cost, divide by the number of patients, you get $100,000 to $200,000 per patient. And IA studies and fungal studies tend to be big. Johan Mertens also shared this slide at a recent talk, 4.5 years on average to run the recent IA studies. Context, complicated UTI trial of the same size, 18 months. These pre-COVID, these days, all bets are off. Next learning objective. There are alternatives to non-inferiority randomized controlled trials, but they're little used in anti-infectives. So let's look. This is from a talk that Rob Hemmings gave at Trends in Medical Mycology at Aberdeen this last fall, super talk. So he says, look, when do you not have to do an RCT? Well, there are times when you don't actually need to. These are the settings where the treatment effects are unequivocal and the magnitude can be understood without a concurrent control arm. So you need something where the treatment is entirely, where the effect without treatment is entirely and accurately predictable, like progression of some of the neuromuscular diseases of childhood. And if you're lucky, you may even have an external data set so you can line up against that. More on that on the next slide. But it's the idea that there are settings where it's very strong. You don't have to. There are settings where you genuinely can't, and those occasionally occur. And this is where the idea of an underpowered RCT comes up. We'll talk about that more in a second. And then I don't want to, well, that's different. That's not usually an answer. So let's talk about single arm trials. When the outcome without treatment is reliably predictable. This is an idea that you see in ICH E10, the idea of the patient as their own control, or actually the counterfactual of what would have happened if you had not intervened. So the disease has to be entirely predictable. Tumors don't go away. Endocarditis does not spontaneously cure itself. You may also be able to provide some external control data to line up against it. So it's not just the patient as their own control, but also the patient versus some external control data. It gives you a little context. Those two ideas are mutually complementary. Both approaches are biased. You have to deal with that, but it doesn't mean the data are uninterpretable. And you're not going to see a lights on, lights off, 0% versus 100%, but a large effect can be compelling in a setting with a really obvious counterfactual, that what would have happened had you not intervened. There are some diseases where you just know that the next thing that's going to happen is not good. This is an idea that's been batted around for many years. Well, I'd rather have an RCT than not. Some randomized data are better than none. The underpowered RCT. Okay, so you won't take an alpha of 5%. How about if I come in with an alpha of 20%? Are you sure you really want me to come in with a trial that's that small? Does that feel like, sort of like we've adjusted the boundaries to deal with what we can do? And also there's this asymmetric risk thing. The sponsor has the problem that in a really tiny study, the study can easily fail due to noise. You've had 10 patients in each arm of a 20 patient study. You could show superiority with that if it was 100% versus 0, but it's probably ain't going to be. And it doesn't take much heterogeneity because every patient's worth 10%. The regulator worries about the risk of approving an ineffective therapy. The other idea that gets thrown in here sometimes is go Bayesian, borrow data from elsewhere. Borrowing data from other RCTs or from experience at other body sites would seem entirely logical, but none of us have ever gotten happy with how you weight the external data in doing that. It's just, it's never happened to my knowledge. An idea that I think there is a little bit more room on is that of extrapolation. And here I'm going to point out a recent paper by the EMA where they thought in detail about the idea of extrapolating. And I like the way they describe it. It's about the idea that you extend information and conclusions available from studies in one or more subgroups to make inferences for another subgroup, and thus reduce the amount of, or the general need for, additional evidence generation. And in fact, we do this all the time. IDSA's expert treatment guidelines, when we adapt for children, there are other examples. In a way, the idea that these ways you can get approved with just one RCT is a form of extrapolation. And I think we need to start being more systematic about this for situations that can't readily or ever be studied in adults. This feels like a fertile ground for community consensus. So our last two learning objections deal with the issue of broadly community consensus, because some of the choices about what it is that we require for new agents to get approved are a function of how we have agreed, what we're willing to accept. So this is about the limits on the data. And let me start off by saying something that may sound a little surprising. I sometimes hear the phrase regulatory hurdle, and I've been guilty of saying that. I have to say that's not great language, because the desire for better data is universal. It's really just the case that the regulatory agencies are the first ones to look at your data and say, well, explain this to me. Why would I want to be treated with this stuff, much less anybody else? So it's tricky when it does feel like that's the speed bump, and yet it's not just the regulators. It's going to be the payers and everybody else. And when you hear the phrase, well, we need an RCT, how many papers have you seen that ended with that? We need a randomized trial. I want you to think about this talk and think about feasibility and think about what can you learn know about the natural history of an infection. Are there alternatives to RCTs? Think of that Rob Hemmings talk from Trends in Medical Mycology. And let me say, there's no easy fix for the challenge of rare pathogens. And that's why I was asked to give this talk, I think, because fungi fundamentally are rare pathogens. Essentially, all of them, I think, are orphan drugs. Orphan diseases. And the data for rare pathogens are expensive in terms of time and money. Some fungi are too rare to ever be studied in an RCT of greater than 3. I mean, you're just, how often are you going to get one of those fungi? RCTs are not perfect, by the way. They're always limited to the subset of patients of interest, only the people who consent at your sites. And remember the problem with moving patients to a master site. And superiority is not usually a fix. No one wants to be randomized to obviously inappropriate therapy. We always want to try something, even if it doesn't look very good. Think about what happened during the COVID outbreak. For how long were people willing to be on a placebo-controlled study of a COVID therapeutic? About 2 microseconds. So we as a community have to engage on this. And the relatively rare nature of fungal infections makes this particularly critical if we're going to develop new antifungal therapies at all. So Maurizio, I want your compounds to succeed, and you're going to whack up into this problem about 10 years from now. What trial can I do? And you'll be frustrated because it's tough. And the core question is, what's enough data to establish risk benefit for these relatively uncommon orphan drug frequency infections? And with that, I stop and I thank you. Oh, wait, I'm sorry, not quite. Look in the deck, there's a slide that's got a bunch of resources if you want to read more of this stuff, root around in there. And I'll make a plug here for the AMR.Solutions newsletter that I fell into publishing 3 or 4 years ago. It puts out stuff that regularly that's of interest to the developer community. It'll feel mostly antibacterial, but really antifungals fall in exactly the same pattern as antibacterials. So thanks for listening. Back to you, Pete.
Thank you, John. Thank you. That was a really nice review of some of these challenges, and once again going through the rationale of the non-inferiority type of design. I think we've been doing it for so long, and many of us still don't understand the why.
Yeah.
I want to ask you to categorize this in terms of the context of your talk. Let's take I think one of the challenges that we've encountered in antifungal trials, so let's say in candidemia studies, has been focusing on one pathogen. So for instance, I mean, candidemia is 5 or 6 different diseases. It's not really one, and yet we clump them all together. But let's say that we want to focus on Candida glabrata, an agent that obviously gives us more fits than most, excluding Candida auris. And let's suppose that we could identify 50 to 100 patients with Candida glabrata trying drug X, and we're able to enroll them in a single arm or maybe even a randomized type of trial. Would this fall into the category? I'm trying to remember was it LPAD or LAPAD, or would that fall into that category of rare pathogens that the FDA would be, or the EMA would be willing to accept fewer numbers in order to, get at a pathogen that's important, but you've demonstrated with a reasonable degree of certainty that you've got an effective agent?
Well, that slide's worth looking at again. So let me just pop it back on the screen. So I would like everybody to know about LPAD. This was created in 2016 as part of the 21st Century Cures Act. And it was an idea that, in a way, FDA doesn't need this. FDA does this all the time. They always consider the severity, rarity, and prevalence of the disease under study. And there are drugs that, what's the smallest data set with which a drug has ever been approved? The answer is N equals 13. It was a genetic amino acid metabolism deficiency, and the therapy was something that was just, it was so obvious, so you could actually get a square wave effect in a real human being with starting and stopping the intervention. So 13 people. So there are times when an itty bitty tiny data set is adequate. LPAD is the specific formulation of this that for antibacterials and antifungals. Congress said, we recognize that these are hard because they're not as sharp-edged in general as a metabolic disease. And that's actually the problem with candidemia or systemic candidiasis, is that the disease has multiple variants, and there are some interventions that kind of work a little bit. So you pull out the catheter, that's the nidus. You still need to treat this person, but you've actually already made a big jump. It's a little easier to think about cryptococcal meningitis. Have you ever seen anybody with cryptococcal meningitis spontaneously get better? No.
No, no.
They don't. And it's the cryptococcus is almost one disease. I mean, you can have other things, of course, cryptococcal pneumonia. There are other body sites, but by and large, the cryptococcus is the meningeal syndrome. And there you could almost imagine a single arm trial with the cryptococcus that would be compelling. If I showed you a therapy that I said, look, a year out in 50 patients with the cryptococcus, the mortality rate's been 3%, and 60 of them are completely without symptoms. You say, wow, that's not what I expected to have happen to those people, right? It wouldn't take a lot. And we actually do use an absolute performance standard for drugs for streptococcal pharyngitis, believe it or not. If you want to develop a new drug for strep pharyngitis, what you have to do is show I think it's a 95% elimination rate of streptococcus out of the back of the throat. I mean, it's an interesting example of that flavor. So I'll flip the question back to you and say, how much is enough? That's the question. What would convince you if I said, Pete, you've got Candida glabrata in your blood. How much data do you want to try a drug other than the kind of Candida? I mean, you tell me, what does it take? And so there's not a perfect answer to this. It's a little bit like pornography. You kind of know it when you see it, but it's got to be a strong signal. And that's also why I made this point about where's that slide? The slide about Rob Hemmings, this one right here. When there's a really strong effect. So endocarditis, how many cases of endocarditis do you have to cure before you believe something works? Not terribly many. Now you may need a few hundred people for your safety database. That's a different story.
Yeah. Yeah.
So, I mean, there's no perfect answer to this, but the big theme has to be one of, if we always are going to insist on a large randomized trial for every aspect of this, we won't make any progress.
Well, that's an important point. It's a very important point. And then Karen Schlam just indicated or chatted that the LPAD approval is usually a condition that an ongoing larger phase 3 trial for efficacy and safety are done following the approval, especially if the approval is limited to, and that's a reasonable expectation, I think, probably.
Yeah, you'd think that if a larger trial can be done, you ought to do it. But think about the EMA's 3 tiers. I think that's helpful as well. EMA recognizes there are settings where you approve initially based on limited data, and more data can be generated, but it's going to take a long time. And there's already this. I like the LPAD language. There's already this small limited population. They need it today. And they're going to say, the fact that you don't have more data is fine with me because I'm otherwise going to die. I'll take the risk. But then the larger population wants the greater data set. So if you can do it, you should. But then remember, EMA's also got the exceptional circumstances where they say, you've done all you can ever do, and we get it. And there's no point in jumping up and down and saying, generate more data when you know it's never going to be possible. And I think that it's got to be a balanced conversation. Every one of these studies there's not the LPAD legislation doesn't mandate another trial. Unlike accelerated approval, where it does. Accelerated approval, you're required to generate more comprehensive data if you to do that confirmatory study. But LPAD doesn't mandate it. That's a decision that has to be made about what can and cannot be done.
Luis asked a question, then we'll move on. How can our group, MSGERC, how can we help overcome some of these issues?
Well, this is where this idea of community consensus builds. You guys have already done one fabulous thing, which is 15 years ago, you got galactomannan recognized by the FDA as a diagnostic tool in IA.
Yeah.
And, we had this great workshop with the FDA back in 2019, the last time we were permitted.
Whenever it was, pre-COVID.
Pre-COVID. And I think that there is fertile ground that is coming. COVID, I don't want to say anything good at all about COVID, but it has pushed us all into understanding the need to develop new antimicrobials. My favorite phrase, I can't get away without saying it, the antimicrobials are the fire extinguishers of medicine. You got to have them available before the fire breaks out. And so if we want to have these tools in advance, we have to come up with ways to enable them to be developed in advance. And it can't be based on requiring superiority data for everything. It can't be based on requiring a randomized controlled trial for everything. Those things aren't doable. They're not consistent with developing a new drug. And so, maybe this, what I hope, FDA has actually been very interested in this question of how do you advance these questions? And I'm, looking forward to future workshops, and you can turn up at those workshops and say your say as academic docs and say, we're in trouble and let's solve this problem. And be careful when you say, and I need an RCT. Just want you to think about what that means, about what you might get or not get.
It's too easy to fall back on that statement.
It's too easy.
It's far too easy to fall back on that statement. John, thank you so much. As always, appreciate your insights.
My pleasure. Great day. Thank you for having me here.
And so our next speaker, Andre Speck, who is associate professor at WashU, Division of Infectious Diseases, and he's going to give us, I think by necessity, a fly-through review of the 15 new they're not 15, but there are a lot of new antifungals, and Andre is poised to give us a review of these compounds that are in development, not yet approved, but most of these are well into clinical trials, with a couple of exceptions. Thank you, Andre.
So I thank you all for inviting me. I just wanted to let you know I am gonna cut back on some of these because of time. Otherwise, I'd be doing about 30 seconds per compound and not doing any of them justice. So I'm going to be focusing more on the ones that are getting closer to actually being used in patients outside of trials. So, but if you do want to get a good handle on all of them, I would suggest checking out our review, which kind of has the same title from OFID, which we published, I think, in early 2020, which now feels like 25 years ago. So we're going to talk real quickly about why do we need new antifungals, and this is a topic that's going to be not surprising to anybody in this group, which is the mortality is a few things, but one of them is that the mortality is still very, very high in fungal diseases and really doesn't appear to be going down all that much. There's also current deficit of a historic low rate of drug development. And in the 21st century, so this century, we have one new novel class of antifungals, which was the echinocandins, which were approved in 2001. As of now, 20 years ago, was the last novel class. Then we got echinocandin number 2, echinocandin number 3, which are effectively the same drug. 2 new azoles, which the big addition there was oral treatment of mucormycosis. But in reality, it's another azole, a drug class that we have had for decades. And we've had 3 new formulations of azoles, but again, these are, while welcome and appreciated, are also just drugs that we've already had. So what does this leave us with in the landscape of treating invasive fungal diseases? And it's 3 classes of antifungals that are really doing the bulk of the work. You know, we sometimes use terbinafine and a few others, but in reality, it's 3 classes. They only target 2 pathways. Only 1 class is oral and 1 is highly toxic. And the oral one is still pretty toxic and has lots of drug interactions. So the deficits that we have also are intrinsic resistance. So there's a lot of fungi out there that do not have any PO options, things like Candida, many of Glabrata and Cioris isolates. Azole-resistant Aspergillus really ends up with IV-only therapy, which is really terrible because you're treating these patients for months and sometimes even years. Intrinsic resistance to all antifungals is present in isolates of many fungi, like Scopulariopsis or Lamentospora. There's also a growing resistance around the world with the emergence of C. auris. You know, there's been some really wonderful data from Duke showing the rise of echinocandin resistance, as well as the rise of azole resistance, more prominently in Europe, which appears to be associated with agricultural azole use or azole-like compound use. So the good thing about this is that we do have a pipeline. And this is the most robust the pipeline has ever looked. And this slide at this point is a little bit old, and we have a few things that have progressed past where we have them on this in this image. But the sheer number of compounds that we see on this image is really what's exciting. And again, I'm gonna focus more on the ones that are up top and closer to the development. But if you do want to see them all, again, our review is quite exhaustive. So cell wall active agents. So we have rezafungin, brexafungerp, and fosmanogepix. Rezafungin is our newest echinocandin that is in development. It's a derivative of anidulafungin. It has increased stability, which may lead to less hepatotoxicity, possibly. We haven't seen the phase 3 data yet, although the phase 3 has completed enrollment, and we should be expecting that data pretty soon. But the real benefit of rezafungin is that we have actually a much longer, much more stable compound. And so the dosing becomes once a week. So instead of having to do daily dosing, we have once a week dosing. It achieves very high levels. You can see here in the PK data on the right. It doesn't appear to have the same paradoxical reactions as caspofungin, so that's good because even given the levels that we're achieving. And it's active against most echinocandin-resistant isolates, and it appears to overcome the resistance largely through just brute PK force. And this does include things like Candida auris, which is the one that we worry about the most long-term, probably. Ibrexafungerp is another compound that has actually recently gained approval for non-systemic disease. So this is an oral beta-glucan synthase inhibitor. It's not an echinocandin, but the way it's often described is an oral echinocandin because it targets the same protein as the echinocandins do. And so it is often called an oral echinocandin, but you can see that the chemical structure is completely different. It has broad activity, very similar to echinocandins. It's active against echinocandin-resistant Candida. And IV formulation at this point doesn't appear to be likely to be marketed anytime soon, so it is an oral-only compound. It's active against Candida, Aspergillus, PJP, dimorphic molds. Those two more in vitro. We don't have a lot of clinical data on that yet, and as well as things like L. prolificans. It has a very high volume of distribution and high tissue penetration, so that's great. It concentrates inside of acidic tissues, which makes it helpful for things like vaginal secretions, which are pretty acidic, as well as abscesses. So a lot of us are really excited to see what's going to happen when we start giving ibrexafungerp to intra-abdominal abscesses, which are highly acidic and would lead to trapping the molecule and actually high rate of very high PK in those abscesses. It doesn't have any penetration to the brain or eye, so that's an important limitation to know about it. And its current areas of study are in invasive candidiasis and is add-on therapy for aspergillosis, kind of following the MAR 2015 study, as kind of trying to look for improvement in invasive aspergillosis. And in VVC, it was actually, for VVC, it was approved already in 2021 under the name Brexafem. And so, but be really careful of dosing. Just because it's available on the market right now for VVC, doesn't mean that we'll be able to use it off-label for invasive disease, because we really don't have the data for that. And the dosing is so different that you'd basically have to be opening a whole package every single day to dose the patients. That would be very difficult. The other, so likely future roles, it's probably going to be involved in step-down therapy, especially in resistant isolates, and possibly azole synergy in aspergillosis, or maybe treatment of a few of the other more rarer things we see. Fosmanogepix. Now, this is the first one that we're mentioning today that actually has a novel target. So unlike the last two, which we talked about, which attack the beta-glucan synthase, this one attacks GWT1. So it's an inhibitor of GWT1, which is a GPI anchor protein synthesis protein. It unmasks fungal cells to the immune system is one of the mechanisms of action. It also destabilizes the fungal cell wall. And it makes it more difficult for it to attach and invade tissue. So it's actually kind of working at both a site of stability, a site of protection from the immune system, and a kind of a virulence factor. It appears to be pretty low toxicity because it has no activity against the closest human analog, which is PIGW. It's both PO and IV. This is the compound that was earlier on was mentioned to have been bought out by Pfizer. So Amplix was bought out by Pfizer. As a result, it's now entering phase 3 trials. It has a bunch of orphan drug statuses, and it has some really nice, interesting properties. One, it's got greater than 90% bioavailability. It's pretty broad spectrum. It goes into ex-Candida, Crypto, Cocci, Aspergillus, Fusarium, Mucorales, Limitospora. It lacks activity against C. cruzii. So this is a really important compound. It does not have any activity against cruzii. It is active against fluconazole and concandida-resistant isolates. It has extensive tissue distribution, and most importantly, it does get into the brain and eye. So this could be our future oral compound for use of things like endophthalmitis with fluke-resistant strain, things like that. And it eventually is excreted through the biliary tree. Future roles, broad antifungal use, trials ongoing. And then next we have cell membrane active agents, which I'm literally just going to go very quickly through because they're relatively early on, and I really don't want to go too far into them. So the first one is azoles. We know azoles. We all love azoles. They're our workhorses. They're kind of the thing that we probably prescribe the most. I think the azoles at this point are about 70% of my prescriptions. And so the next generation azoles, which are made by Biomed, which has now been bought out, are really interesting. And I'm not going to belabor their point, going too much into them because they are pretty early on. But we often refer to them as intelligent designs. These are azoles that have taken all the things that went wrong with azoles and tried to make it better. So what they did is increase binding to CYP51. So they actually bind much better to fungi, which should increase lethality. They much lower binding to the mammalian CYP450 system, so they should make it less toxic and less drug-drug interactions. They have very long half-lives, greater than 6 days for all of them. They stay around for a very long time. They have good absorption and very predictable PK. So these should be the next generation of azoles, hopefully a very forgiving azole. The next compound we're going to talk about, again, in this like shotgun approach to antifungals, is going to be the inculcated amphotericin B, or MAT 2203. A compound I'm personally very excited for, because one of the things I often tell my residents is the biggest determinant of how well people do with many fungal infections is how much amphotericin we can shove into their body. And so inculcated amphotericin B uses this very cool technology of lipid particles, nanocrystals, which trap the drug and make it orally bioavailable. So the drug gets taken up through the usual chyle system and then dumped into our blood, where it still stays inside of the sheet and is protected until it is phagocytosed into a low, I believe it's a low pH and low calcium environment. But I would have to be corrected by the actual people who know this compound better than I do, which I'm guessing are on the call. And what's wonderful is that it's the antigen presenting cells and cells that engage phagocytosis in humans and fungi that will actually eat this and then open up amphotericin inside of the body. And so they're delivered to the site of the target, which allows us to push the dose quite a bit higher so that we may actually be able to get much more amphotericin into people. The kidneys appear to be protected, which is always great because I am the #1 enemy of our nephrologists. And it doesn't appear to change the spectrum of activity. We're starting to get really early human data now. I'm really excited about the future of this compound. So the next part is big, and this is we're going to be talking about intracellular agents. But again, for the interest of time, we're going to be kind of ignoring a lot of these. Many of them are kind of early in their development, and some of them are not progressing at this rate, and some are going to be progressing in the future. So, but again, for the interest of time, we're really going to be only talking about olorofim. The other ones, again, if you want to learn more about them, there is the review, and I would really strongly recommend. It's a really good primer. So if we talk about olorofim, it is a DHODH inhibitor. So it inhibits DNA synthesis, and this is a very much an oversimplification, and John, please forgive me. I often tell my residents and fellows to think of this as think of it as a fungal Bactrim, right? So that's kind of how I think of it. It inhibits DNA synthesis. The binding against the mammalian analog is much lower, so it really doesn't have any activity. It's orally bioavailable. It has wide tissue distribution, including the brain and synovium, and has orphan drug status against things like Lamentospora, and it's become the drug that through a trial I've been using a lot for salvage. It has very good activity against hyaline hyphomycetes, dematiaceous molds, dimorphic molds. It's active against highly resistant strains, so things like Scedosporium, Lamentospora, Scapular Epsibrumtii. And it has no activity against yeast and mucorales, which is actually kind of nice from our standpoint, because if you have, if you had a pan antifungal, it may become the most overindulged drug by the BMT service. Resistance appears to be really hard to induce. Hepatotoxicity signal is comparable to azole, so it does have that kind of a signal. It's currently in an open label salvage trial for susceptible molds. Phase 3 trial for Aspergillus is starting soon. Possible clinical roles for things like pan-resistant molds, Aspergillus, dimorphics. I think we're going to see a lot more to come from this compound in the near future. So this is my summary, and with this slide, I will kind of stop talking like an auctioneer. So currently there are limited options for many fungal infections, but what's really amazing at this moment in time is that there is now a salvage option for every single patient. So what that means is if you have a patient who's completely failing or a patient who has a pan-resistant pan-resistant mold, you actually do have, at least through a clinical trial, a salvage option for them. Whereas in the past, for a lot of these, things like Lamentospora and some of the other ones, we would see the patient would say, well, call palliative. So I'm hoping that that is the future change that we see in mycology. And so with that, I will stop.
Thank you for the nice review, Andre. They're good. We'll start out with a couple of questions that came through while you were talking, and then I have one specifically for you. One of the questions was the ibrexafungerp plan moving forward. First of all, it's really tempting when you have patients with refractory Candida, stomatitis, or esophagitis due to resistant organisms not to prescribe this agent when they failed other agents. I think we've been challenged with that on a couple of occasions. Thankfully, we've been able to get them on the Fury study, and,
right.
And that's been a nice kind of way to get them enrolled. But a question came up about a candidemia trial, and I just responded by saying that there is a candidemia trial that has been designed. There are probably a few little wrinkles that need to be ironed out, but that trial is designed to, or is designed with the intention of moving patients who receive Ibrexa over to oral therapy very quickly. Do you want to say anything about that?
Yeah, so I think that's going to be, it's a nice trial because the idea is very pragmatic, right? Because it's the role that it's probably going to fill is going to be that transition to oral. So it's going to be, I think, in the very near future, starting to enroll.
First quarter is what we've been told.
Yeah, that's kind of what I've heard. And the idea will be to, very early transition as soon as the PIs start to feel comfortable, even as early as day 3, to transition from IV echinocandins to a randomized fluke versus ibrexafungerp, assuming that they're susceptible, right? Because you're not going to be transitioning to fluke if they're fluke resistant.
Well, I mean, we'll hear a little bit more about this. Joe Vasquez is going to talk to us about, early or de-escalation types of trials. Right. So this kind of broadly falls under that category.
Yeah, and I think what I wanted to say early on, but I didn't, is that the LPAD process that John made a comment of is largely a big part of why we're seeing this antifungal renaissance. It is the thing that is allowing it to become more commercially viable. And so it's part of why we're going to be seeing this now.
Okay. Jay Aram, who works at Pfizer, has an organism specific question, and I'm going to follow that up with another one. He says, it doesn't seem that any of the antifungal development really address the mucorales. Is that correct? And if so, why do you think that is?
Mucor has just historically been a difficult compound, or difficult organism. And I think, to quote our friends in surgery, for mucor, nothing can heal like cold hard steel. And for mucor, one of the most important things always has been surgery. But there are compounds that do actually have mucorales activity, the azoles, the amphotericin, the oral amphotericin, presumably should have activity. So we will be seeing some of it. Hopefully soon, so the issue has always been very difficult research field. It's very hard to enroll enough mucor to get a clinical approval for mucor.
All right, well, the follow-up to that, another organism specific question is, as I've looked at these compounds, I've not seen anything that just really blows me away with its fusarium activity. Now, I know there are a couple of organisms or a couple of the species that olefin picks up, but what have you seen as it relates to fusarium? Because we talk about, the big three, fusarium's really always in there. So.
Yeah, fusarium is a difficult one. The compound that I think is interesting for olefin does have very mixed activity. And if you get a good MIC, you maybe you'll do okay. I think we actually did have a fusarium into the trial in olefin. However, the compound that appears to be getting some of the more play with fusarium has been phosphomatogepics. And they published, I think, in ECMID last year, kind of a salvage from their EIND with about 70% success rate with refractory fusarium. So that's partially, that number sounds really great to begin with, but it's refractory and it's EIND. So a decent amount of people died by the time they got to that stage. And so then maybe you're selecting a little bit more of a safer group, but it does have very good activity against fusarium, and so I'm hoping that is a venue where fusarium gets better coverage.
Yeah, we remember that voriconazole got approval for fusarium on the basis of about a success rate. If I'm not misquoting, it was like success in 7 out of 13 patients.
Right.
It was. Yeah, these are very difficult. Neil Stone asked this question, great summary. And he wants to know, are there any available published phase 2 data on olorifim? Hard to convince my hemonc colleagues to get on board with a phase 3 without first seeing the details of that. I know there are phase 2 data, but have we
seen? That might actually be a much better question for John. But I will say that, the phase 2, the single arm phase 2 is going well, and it's about 3/4 enrolled right now. And there's some obscene number of patient years of experience at this point for toxicity. And so toxicity data looks pretty good. The difficulty is always, individual numbers, but anecdotally, we're seeing really good responses. And but I will yield my time to John.
John, would you care to speak to the phase 2 data and where it's at and what kind of patients when it'll be published?
So I don't want this to be an olorifim infomercial. Let me just remind you, olorifim has breakthrough therapy designation from the FDA twice. We got it initially for invasive molds, and then we got it again for CNS cocci. And you can infer, this based on preliminary clinical data, that it did something that nothing else was doing. And we've not yet shown the data from the ongoing phase 2 trial because it's an ongoing phase 2 trial, and we're trying to get to the end of it before we put it out there. Because if we release the data, then we're going to start to influence study conduct. Who you choose to enroll, why you keep somebody in a trial might be changed by the fact that we release the data. We've put a few case reports out. And I say breakthrough therapy designation, you can get the wording of it off of our website. So let me just stop there and say it's a slippery slope when you start to disclose data from an ongoing trial, and that's the essential point.
Okay. All right. Thank you, John. Okay. Are there any other questions? Because if not, this is a good break time, and we may have found ourselves 5 extra minutes here. So. Andre, I would like to thank you for presenting that. I think that you've really, you and your group have distinguished yourself for putting together what I think is really the best review of the new antifungal agents out there. It's very complete. It even includes Aereo basidium and a lot of other agents that may or may not see the light of day, but still it's very complete. Gives really good timelines. That graph is something that I like to use and talks that I give on this topic. So
it's a funny story because Paul Sachs invited me to write it based off of a tweet, and then when we wrote it, I sent him an email saying, "Hey, it's 9,500 words. Do you want this to be 2 or 3?" And he said, "No, we'll just do it all as one big one."
Well, it's encyclopedic.
It's encyclopedic, yeah.
All right. Well, Andre, thank you. thank the other speakers that we've had thus far. We're going to take a break, but we will start promptly at a quarter of the hour. Our next speaker will be Elizabeth Dodds-Ashley. She's going to talk about antifungal stewardship, and we're looking forward to that. But we'll see you in 15 minutes. Thank you. Okay, it's 1:45. Then we'll go ahead and resume the second half of our program. I would like to welcome Libby Dodds-Ashley, another good friend from working together for a long time. Libby's PharmD, associate professor in the Division of Infectious Diseases at Duke University. She is going to talk to us about antifungal stewardship. Libby's interest is it's not exclusively antifungals, but she has written extensively about this, and really, really pleased to have her here. She's quite experienced and looking forward to what she has to say. Thank you for being with us, Libby.
There, can you all hear me now?
Yep, and your screen looks great.
Great, thank you. Well, thank you for that nice introduction. I'm excited to be here and see you all virtually. Sorry we can't be in person. And I hope to share some exciting work that I want to right out of the gate acknowledge my colleagues, Russ Lewis and Melissa Johnson, for their very hard work on this effort. We have all been working in the area of antifungals for most of our careers, and especially Melissa and I have done a lot of work on antifungal and antimicrobial stewardship in the recent past, and this is our attempt to merge those two worlds. So in order to really cover what's new and some current gaps, I wanted to take a moment to go back and talk a little bit about antimicrobial stewardship, which I think is pretty widely known as a practice, but I did want to spend some time on the frameworks that we build a lot of these practices on. And it has to do with a set of factors we have come to call the core elements. These were first released by the US CDC, and you can see the current core elements from the US CDC on the right half of your screen. And they were followed by an international consensus document more recently that was published in 2019. It was the result of a modified Delphi process from several different constituents that has largely been adopted now by the World Health Organization to underpin some of their antimicrobial stewardship work that they're doing. These core elements are important, and some of them might seem just accepted practices, but I'll just quickly go through them, and then we'll talk about how we pivoted and made these apply to antifungal stewardship. Key to all of this, I'm not going to talk much more about this today, is having support from senior hospital leadership for your stewardship activities. There needs to be accountability for who's going to be responsible for stewardship. Even though everyone's involved, there does need to be sort of a clear leadership structure for this. You need expertise in managing the infections and also in the drugs that are involved in treating these infections, be it antibacterials, antifungals, and we'll obviously pivot more towards antifungals in just a minute. There needs to be great education and not just mandates. We need to have actions, and we'll talk about those in general and related to antifungal stewardship, as well as monitoring and surveillance of your work, and then reporting and feedback back to frontline prescribers and others. And so with the release of the US CDC core elements, just to show you what having these guiding principles can do, the CDC has been able to track through annual hospital surveys that when these were first released, less than half of US hospitals complied with all 7 of the core elements. And these are for traditional ASP programs, and I'll show you in a minute how many of those may or may not include antifungal stewardship at baseline. But as you can see, with time and with attention, and a few requirements related to CMS, but not as many as you might have thought. They've actually gotten to well over 90% adherence with all 7 core elements with the over 4,000 hospitals in the United States today. So just having these out there and then trying to monitor them with time can really move the needle in terms of having these elements that we consider so essential to using antibiotics appropriately and causing minimal harm and preserving them for the future has really had great success. So that leads us to ask, I think, and I will shame myself, we are guilty here. When I was the pharmacist lead for the Duke stewardship team for many years, we often left antifungals for the last area on our list. And there was a group through the Shea Research Network that in 2018 actually conducted a survey of the members of the Research Network facilities. So remember, this is not all US hospitals, much smaller sample size, only 111 hospitals surveyed. A little under half of them responded to see how often their ASP, which at that point was required to be in place, actually included antifungal work. Now, I think surprisingly, but again, this is a very select survey population. 96% of them actually included antifungal stewardship as part of the routine work of their stewardship team, but not all the core elements were there when it came to the antifungal drug use. So we look at our 2 key actions, either being prospective audit and feedback to prescribers in real time or having some guidelines for treating common fungal diseases. Those occurred in almost 3/4 and just about half respectively. Rapid laboratory testing was available in 71% of hospitals, but this was much more variable when you looked at practices such as antifungal susceptibility testing available locally and universally for bloodstream isolates, just as an example. Also, mechanisms to perform surveillance of antifungal drug use. So measuring the antifungal drugs they use only in 64% of hospitals at this time. Remember, this was just conducted in 2018. And then only about 1/3 of them report that antifungal drug use into the CDC through the National Healthcare Safety Network. And we're going to talk about that a little bit more just to give you some ideas of antifungal tools that are out there for stewards to help implement antifungal stewardship practices. So knowing that we had come so far in antimicrobial stewardship and knowing that we had big strides to take in antifungal stewardship, we worked with the MSG to develop the core elements of antifungal stewardship. And this was published in August of 2020. Melissa Johnson is the first author on the paper, and it's in a supplement to JID. I will say that this graphic does not appear in that publication. That is from a publication just last year that came out from Payal Patel that talked about some additional challenges of antifungal stewardship in immunocompromised populations. So I thought they did a great job with this graphic, so I decided to use it today. And we were very easily, through a lot of discussion and hard thought, able to apply each of the core elements to specific examples of how we could optimize antifungal use in our hospitalized patients today. So using the same principles of the international core elements that were developed through that consensus process, we pivoted and released these antifungal stewardship core elements, which really can guide ways to have a robust antifungal stewardship program. We have a brief time today, so what I want to do is actually highlight many of the questions that I get from people about having antifungal stewardship programs. And in order to do that, I think it's important that we set the stage about what we mean for antifungal stewardship. I know there's a lot of colleagues on here who are very involved in drug development and designing studies for drug approvals and optimizing and defining patient populations that will most benefit from antifungal drug therapies. But the reality is the group of people on this call are not the primary users of antifungal drugs in US hospitals today, and those need to be the targets for the stewardship program. Just as perspective, I currently work in a network of community hospitals, and so I took the liberty of using some of our data. And when we look at all of the admissions in our 26 community hospitals, 56% of them will get an antibacterial drug during their hospital admission. And that's a lot. And you can see how that's a big target for traditional antimicrobial stewardship programs. When we pivot that and look at those same admissions and the percent that will receive a systemic antifungal therapy, it drops to only 3%, which may not seem like a lot. However, you need to think about this. Our hospitals in our network, our average bed size of 150, we go down to 20, we go up to a little over 600. But in that hospital sample, we still give more than 7,200 patients just last year alone a systemic antifungal drug. That is a lot of patients. And on average per hospital, it's 277 admissions a year, almost an admission a day, and over 1,300 days of antimicrobial therapy, antifungal therapy that are given to our hospitalized patients in the community setting. And we'll talk about what resources are available to them, but we need to think about antifungal stewardship across the board. There is a lot of antifungals use in areas that are not seen by ID practitioners, that are done in community hospitals or in areas of academic medical centers that are not heavily presenced by infectious disease specialists. So now let's pivot with that bit of perspective and think about some questions that have come up and ways to frame your program. And I think the one I most commonly get from people and colleagues is, do I need to have a standalone antifungal stewardship program versus integrating antifungal stewardship into existing stewardship practices? And within our guidance document that we published, we had a few recommendations. And if you haven't had a chance to review that article yet, this is the framework we use. We go through each of the core elements, and then under each have key recommendations for practice related to antifungal stewardship. Our best guidance is, when possible, to incorporate and leverage the expertise of the local stewards with the expertise of your mycologists and the ID physicians who specialize in treating patients with fungal disease. So many stewardship programs will not have that antifungal or fungal disease management expertise embedded within them. And so it's a great opportunity to leverage those different areas of specialty in order to optimize various ways to optimize drug use. And I'll show you some examples of that as we go through this. But there is no need to reinvent the wheel when it comes to ways to fine tune and optimize therapy. Also, we don't want to be bombarding the primary teams in care of these patients with multiple different groups contacting them. So here's your call about your vancomycin, and here's your call about your voriconazole, as an example. So our recommendation is, whenever possible, to try to integrate these activities. The stewards, they've come a lot in the last few decades and have great tools available, and often know how to leverage different systems within the facility, including the electronic health record, to help promote the right way to use antifungal drugs in your patients and move forward from there. So not a one-size-fits-all answer, though. And I will say that if you're a stewardship team and you do not have antifungal expertise at the table, you need to get it there. And I will go back to something I said a long time ago. I think we sometimes think in a very siloed fashion around what that means. Does that mean that you need your group that has expertise in treating fungal infections at your third Tuesday of the month stewardship meeting all the time? The answer is, you might not. You might need to go to those groups. You might need to go to the hematology oncology group as a stewardship representative, and not think of this traditionally as the stewardship team meets when everyone who's on the stewardship committee is at the same table. What it means is we need to all leverage this together. And so it might mean putting a stewardship representative on an existing committee that's thinking about the care, management, prevention, and treatment of fungal diseases in these high-risk patient populations. So it's very important to think a little bit differently in what it is to meet as a stewardship team. There might be multiple different sub-teams of your stewardship team that make it successful at a large academic medical center. A small community hospital, it might be the people you can get together and then bringing in outside expertise that we'll bring up in just a minute. In our guidance document, we also heavily endorse the fact that whenever it is possible, infectious disease consultation should be performed for some key fungal diagnoses. But the reality is, and we see this all the time in the DASON network where we do stewardship today, 63% of Americans live in an area with no ID specialist access. We see this a lot within DASON itself. 30% of our hospitals do not have the ability for an ID consult on site at the hospital. And this does not mean that we should just abandon the need for this specialized expertise. There are tremendous models. I don't have time to review them all today, but through remote consultation, through tele-ID consults, through tele-stewardship expertise, you can actually bring in very, very talented and specialized infectious disease specialists for treating fungal diseases from all over the country and sometimes the world to help guide these practices. And there are new models emerging that include compensation for this. And hospitals are getting more creative in how they bring the experts locally, perhaps a positive benefit of the pandemic and all the telemedicine we've had to learn and pivot to so quickly. So there needs to be that specialist access. There needs to be creative ways to get it to all patients, and it needs to be done within the resources that are available. And that's one of going to be our ongoing questions, is how do we adequately resource stewardship teams to do the antifungal stewardship that is so badly needed today? Another question we get a lot is, what laboratory testing do we need to optimize our antifungal stewardship program? And we, in the article, review several of the available technologies out there and how they can be implemented in various settings. But I just wanted to raise a few items of interest that we've come across, especially in our community hospitals, but we also see sometimes even in large academic medical centers. Just because the technology is there, bringing it in does not mean it will solve a problem. And this is a great way where the stewardship program can work with the microbiology laboratory to optimize both avenues, workflows on the micro side, as well as optimizing treatment decisions on the patient care side. Some key considerations, and I think Melissa's on the line today. I know we've both run into this in our community hospitals where we advocate heavily for having susceptibility testing available for blood isolates of Candida, for example. And we have several hospitals that would check that box as yes. And then when we talked to them, we found out that it was a send out that was taking 7 or 8 days to come back. Well, that was a large expense for the hospital proportionate to what they were receiving in reimbursements. And also it wasn't giving us any meaningful data to help drive patient care. So you really need to go through a checklist of key elements around laboratory testing to be sure it's helpful. Some of the other things to consider after turnaround time are, do you have the local capacity for testing? We see microbiology laboratories all the time that have great diagnostics on the shelf, but only have a few people trained on them that don't have the time to run all the tests with everything else they have to do in the day. And so the start of these rapid diagnostics is delayed by hours, essentially losing some of the utility and tying up a lot of resource and the capital to buy those systems. You also need to make sure that the frontline decision makers are really receptive to the tests that you're bringing in. And I think this is so important. Be sure that they're at the table when you're designing these algorithms and how you're going to deploy this testing, because it may not matter that you have a great new test. It may not be actually something that they're willing to change practice on due to other things that might require some education to reverse or change prescribing practices. You can see some of the other checklist items here. You also want to think about other local practices that could interfere with the testing. There's a pharmacy list served for stewards, and actually just yesterday there were questions out there about some hospitals that were struggling with beta-D-glucan testing in their ICUs, and it was coming up universally positive, and they didn't know how to address that with providers because it was actually leading to an increase in antifungal drug use in that particular ICU setting. So always things to think about. What are the other things happening at your hospital that might make the testing not as useful as it has been in published studies? Now, I do want to bring up one interesting element around laboratory testing as it relates to antifungal stewardship, and I'll keep this brief, but when you might be at an institution where you are very interested in bringing in a certain technology around rapid identification or rapid susceptibility testing for bacteria, but it might be hard to cost justify, even if you can break down the silos between laboratory and pharmacy and even on to patient-centered outcomes like length of stay. There's actually evidence out there that in some cases, because of the higher cost of some of the antifungal drugs, they alone cannot justify those technology platforms. But if it's a platform that tests for multiple bacteria and fungal pathogens, for example, you can sometimes cost share, and bringing in the antifungal stewardship element can tip that over. There's a few great examples of that out there that we can share if you're interested. Now, what about actions? That is core element number 5 in most of the rubrics. And are there best antifungal stewardship practices to adopt? I think that hands down, when you look at invasive candidemia, the bundle approach that has a set of metrics that are used and are protocolized whenever a patient has a candidemia identified have been shown to improve initially, arguably, process measures, I will admit, but you can certainly improve things that are tied to better outcomes for patients. And at the end of the talk, I'll show you some newer data that are emerging that even suggest mortality is a benefit in a recent trial that was published. Also, there are many daily stewardship interventions that are going on, primarily focused on antibacterials. I think one of the ones that's getting the most attention right now are handshake stewardship rounds. These are quick verbal communications between the primary team and stewards, be it pharmacists or physicians who have reviewed the patient, where you can quickly discuss the plans for antibiotic and antifungal care. Because this is already an existing structure, this is a great way to put additional elements of antifungal stewardship. And this is a great way that with backup and support from the experts in treating fungal disease at your facility, be it on-site or remote, you can also expand antifungal stewardship in that existing set of resources without being a lot of extra additional resource that you need to devote. Now, I could talk literally all day about antifungal stewardship interventions, but as we get close to the end of our time, I thought I would just highlight that Russ, Melissa, and I, along with the MSG leadership and others, had categorized these. And this is where we spent a lot of work into having the elements that we thought were essential stewardship interventions to have a successful antifungal stewardship program, having achievable and then aspirational antifungal stewardship activities and goals. And I encourage you to look through these and reach out if we can answer any questions about or clarify any of these any further. But the easiest and most essential are integrating antifungal drug use into the basic components of your existing antimicrobial stewardship program if it's not already there. And then looking to grow and expand that to have additional diagnostics to put better information in the hands of clinicians so they can optimize care of patients quickly, to get them on antifungal drugs when they need them and get them off antifungal drugs when they don't, so we can preserve them for future patients. Now, one of the core elements I discussed was measurement. And so I think this is important to spend just a few moments on. And one question we get are, what should we measure with our program once we incorporate antifungals into our antimicrobial stewardship program? And I admit many of these are aspirational, but I did want to share some of the most recent data on the effects of antifungal stewardship programs on actual patient outcomes. Now, the first and the most commonly cited is not a patient outcome, it's decreased antifungal drug use. And I will completely admit that this alone will probably not motivate many programs to tackle antifungal stewardship with the many complexities that go into managing these patients. However, we now have data from the recently published COMIC trial that when you implement a candidemia bundle, you actually can have improved mortality. Now, I will admit this is a single study with many of these, as with many, the sample size is low, but the fact that by optimizing the process of treating patients with candidemia actually showed a difference in mortality and not just on measures like the proportion of patients with an eye exam is significant to me. And so I eagerly follow it for a larger sample size to see if this is confirmed. There have also been just last year a publication that showed that antifungal stewardship efforts to get patients off of antifungal drugs actually decreased recurrence with resistant disease. And in a small study of neonatal patients, there was actually has been shown that with a very robust antifungal stewardship program that had about a complement of about 10 different interventions all intertwined. That might sound a little more complicated than it actually was, but it was a very comprehensive program that actually decreased resistance when tracking colonization in their neonates in their neonatal unit. So these are all reasons that we really should promote stewardship. So by improving the appropriate and decreasing the unnecessary antifungal use in our hospitals, we can really preserve these antifungal drugs for additional patients. And with these new drugs that we just heard about that are coming, we can have hope that we will be able to use them for now and many years to come once we get them available, by making sure we right size their use in patients who need them and minimizing it in those who don't. And then one last question is about antifungal drug use and how do I compare? So if I can measure my antifungal drug use, and remember, not all hospitals, even in that Shea Network survey, were able to do this, but if you can measure your antifungal drug use, can I benchmark it? And the quick answer is yes. And now in the United States, we have the NHSN antimicrobial use option, which is risk adjusted at the facility level for targeted unit types and does include antifungal drug metrics. Their primary metric they use is a ratio. They call it the SAR, the Standard Antibiotic Administration Ratio. And they have developed this for antifungal drugs predominantly used for candidiasis only. So you will not see comparators there, for example, for voriconazole or posaconazole, but you will for others. And it is a start and a way to look at risk-adjusted antifungal drug use, again, for the targeted units. This is also a topic we could talk for hours on and are happy to answer any questions afterwards about the SAR as it relates to antifungal drug use. So I hope I've quickly shown you some of the data we have for antifungal stewardship, the progress we're making towards having this a more comprehensive component of antimicrobial stewardship programs, at least here in the US. But we have many remaining questions, too many to list on one slide. But I think that we still need to carefully think through and advocate for the resources that are needed in a facility related to their antifungal drug use to make sure that it is right-sized. We still struggle on the antibacterial side, but I think antifungal drugs need to be included in that equation and part of that calculation. We also need to know a lot more on which of the antibacterial interventions for stewardship apply best over to antifungal stewardship. What are our optimal outcomes to track? What should we be measuring? Will the findings of some of these admittedly small experiences that I've just shared with you persist in larger assessments of the stewardship efforts? And then once we determine best practices, what is the best way to educate our frontline providers about this in the midst of all the other information they're having to process and assess for the very complicated patients that are at highest risk for fungal infections? So with that, I want to say thank you, and I think I've left a little time for questions.
Thank you, Libby. That was comprehensive, and you covered a lot of territory in a short period of time. There are 3 or 4 questions I'd like to convey to you. The first is from Don Silla, and we want to know if antimicrobial antifungal trends have been impacted by COVID. We certainly have seen an explosion of use of meropenem. And so the question is, has that really followed along with the antifungals as well?
Yeah, excellent question. We've actually been tracking this quite closely. From the antibacterial side, early in March of 2020, we did see broad spectrum antibacterial drug use, and then by the end of 2021, it was still increased, but it really shifted over to a more community-acquired pneumonia, so a lot of ceftriaxone. And when you talk to the CDC, they're seeing the same trends. A lot of ceftriaxone use in these patients is what we've primarily shifted to. We have seen waves in antifungal drug use. We track it compared to releases and announcements about experiences out there for, with aspergillosis. We've had a few regional cases here of mucormycosis. There's actually a prominent physician who was infected with COVID and ended up developing rhinocerebral mucor that got a lot of attention and actually led to a lot of antifungal therapy empirically as a result. So we are seeing trends like that. We are actually in the process of fully teasing out which of those patients are COVID. We've developed some denominators for that, but yes, certainly COVID is impacting this across the board. And we are seeing in our patients, we're putting some into the database, some cases of candidemia in patients, especially those when we started using high dose steroids at some of our community hospitals. So definitely have seen impacts of COVID.
Right. John Badley asked the question. It's a good one because we seem to be seeing this, but how are you addressing diagnostic stewardship? And for example, regulation of diagnostic tests, and they, like others, are seeing broad and maybe inappropriate use of T2 Candida, the galactomannan, the beta-D-glucan. Those are the 3 that would kind of come to mind. There are others as well, but are y'all keeping track of that? And what are you doing? And what trends have you seen, if you are, and what are you doing about it?
Yeah, diagnostic stewardship is very important, and we see a lot of misuse of diagnostic tests. People may come from an institution where they trained and had access to galactomannan, and so we'll come into a facility and say, I need to have galactomannan here, and they don't realize that they don't even have, more than a handful of aspergillosis cases in a year, so it's not really an appropriate use of the test, or they aren't willing to respond to the test. And so we do see some interesting trends. They tend to be very hospital specific. So what we do is go in and assess the use of a diagnostic and then show them, this percentage of time is how often you appropriately respond per your own institutional protocol to the test that you've ordered. I'll tell you, I don't think it's much different than when we first got antifungal drug concentration monitoring here at Duke. And I used to make the Duke fellows when they wanted a voriconazole concentration, and it was going to cost hundreds of dollars, and we had to conserve that resource with the lab. I used to make the fellows write on a piece of paper and put it in an envelope and say, what are you going to do when that concentration comes back? And I think that's the hardest thing, is getting the data and then making sure we're actually using it to change patient care. And that's where we don't see that happening a lot. And then there are lots of other examples, like the pharmacy listserv I mentioned, where there's an ICU where there's a lot of beta-glucan being ordered, nearly essentially all positive, and they need to get to the bottom of why that is, because it's actually driving up the antifungal use and not decreasing it as the intent is.
Well, we have time for one more question. Louise Ostrowski-Zeichner said, can you provide some practical advice on how to break up the silo mentality that often pits the 3 entities, that is pharmacy, lab, and clinicians, that are all looking for better outcomes, but oftentimes kind of fighting against one another? I don't know if you encountered that there, if you all function as one unit, or if it, if you tend to.
I think that's one of the key reasons that leadership support is so important. And I will say, I came across a very creative model when I was at the University of Rochester that I brought to other places with me. The leadership there was, when they wanted to save money, they just wanted to save money. And so they came to us with a proposal that said, if two departments work together, you both get to credit the savings. And so I've gone to leadership and said that, when there is a test that we want to bring in a community hospital, it's going to cost the lab a little bit, get patients out the door faster, and save pharmacy a little bit, we can sometimes come together and go to leadership and say, look, we want to do this together, but the accounting is what's in the way. And when you come with potential ways to put more patients in bed or save money on the pharmacy side, usually people will listen to you. So I think it's sometimes us breaking down the silos from the bottom up instead of waiting for the top down changes. Because in my experience, when you go and say, well, we have a better way to do this and it's going to increase revenue and it's going to save this person a little bit of money, but this person's got to spend a little bit. Will you let us all be creative? Usually people will listen to you, but they might not come to you and ask you to do it that way. So you kind of have to, knock on the door and say, listen, we have an idea and let us in. We want to take better care. And usually you have success there.
Yeah, I mean, I think a good example is when you bring in a test that might lead to earlier discharges. The lab doesn't receive credit for that. They just see it as a cost.
Right.
Anyway, Libby, thank you again. That was terrific.
Thank you for having me.
Thanks for that. Oh, it's our pleasure. Thank you for being here. Okay. All right, for our next speaker, I'd like to introduce Joe Vasquez. Joe is Division Director and Professor of Medicine at the Medical College of Georgia. It's gone through so many name changes. I'm not going to go there, Joe, but you're going to talk to us about de-escalation trials in mycology. None better to talk about this than you. So
sure. Thank you very much. Can you see my screen?
We can see your face.
Not yet.
Not screen yet. All right.
It's loading. We can see it now. You're good to go.
There you
Wonderful. All right. Well, thank you. Yes. So I've been pushing for this for maybe a decade or so, or maybe more, myself and Shmuel, for many, many years. So it's certainly a pleasure to be here. So today we're going to discuss de-escalation trials. And obviously there aren't a lot of de-escalation trials. So in essence, what I'm going to do is kind of pick specific data and try to get a little bit of information. This talk is obviously a great, the talk before was a great segue into this, because certainly de-escalation is a good part of antimicrobial stewardship. So I've tried to do the best that I could with these. Let's see. All right. Are my slides not? There we go. So conflict of interest or financial consultant for Sadara, Synexus, F2G, and Amplex, and now actually Pfizer. So what are the objectives to this? The objectives today are, one is, what is de-escalation really? Number two are the current de-escalation recommendations, if there are some. Past history of antifungal therapy and de-escalation. Some hopefully future de-escalation trials, and then future recommendations that I'd like to try on everyone. So what do we mean by de-escalation? So I decided, though, I went to Google, and Google gave me this definition of de-escalation: a popular term meaning to withdraw nutritional support and ventilation from a patient in a persistent vegetative state. So I decided, well, that's probably not the right definition for de-escalation. So went to Bing, and then Bing told me it was a reduction of the intensity of a conflict or potentially violent situation. Well, I guess if we're talking infectious disease versus surgery, that could be a problem, but I didn't think that was the one either. So I went back to Google and decided that to decrease and extend volume or scope would be logical and reasonable as far as the definition for de-escalation. And in essence, it is part of antifungal antimicrobial stewardship, really optimizing the four important parts: choosing the right antibiotic, obviously, the dose optimization, and then de-escalation, the route of administration from IV to PO, or starting out with PO, and then the duration of therapy. And certainly shorter is better, as I'm sure everybody. So I actually took this from the paper from Johnson et al. That was published and that was discussed previously. So for some infection studies, meta-analysis have shown that, and compared with longer courses of therapy, shorter courses show similar clinical outcomes with fewer drug-drug reactions, drug interactions, adverse events, diminished length of stay, and certainly cost. So it's very, very important for us to consider this. So this is a list. I'm sure everybody here has seen this. Shorter is better stewardship. And I look at it, it's like, where are the fungal studies? We even have malaria studies, for Christ's sake, and we don't have one fungal study included in this list. So I think we have to do better. It's time for us to do something about it. So what are the current recommendations that we have as far as de-escalation? Well, there isn't anything specifically, but if you go to the clinical practice guidelines that were published by IDSA MSG in 2016, it says transitioning from an echinocandin or amphotericin B, usually within 5 to 7 days, is recommended in clinically stable patients, azole susceptible candida, negative blood cultures. And that was a strong recommendation, moderate quality. The other part is the duration, which still remains 2 weeks after documented clearance of candida from bloodstream. And I guess I'd like to ask, for those of you that have been around longer than I have, is where does these 2 weeks come from? I'm not really sure. The ECMID guidelines for management of candidiasis, they actually have step down on day 3 in patients that are stable, but they also have a duration of 2 weeks. And again, where did that come from? A little bit of historic, I suppose, going back to 2000, the initial practice guidelines had 2 weeks after a last positive blood culture, and then amphotericin to fluconazole, IV to PO, maybe switch. So I guess we have gotten a little ways. The following guidelines that were published in 2009 had the candidemia transitioning from echinocandin amphotericin to fluconazole is recommended for all patients with fluconazole susceptible isolates and are clinically stable. And that was an A2. And the duration didn't change, 2 weeks after the last positive blood culture. And again, I'll ask why. Consider voriconazole on a step down oral therapy for cases of Krusei and some glabrata. So, and this is great, and it's similar to what we have, but the problem is we now have quite a few azole-resistant candida species, specifically Candida glabrata, and we don't have any approved medications for that. So how did we get here where we're at today? Well, John is on the call, so it's great to go back to 1994, one of the initial candidemia studies, which was fluconazole versus amphotericin B. In this study on day 7, there is a possible oral step down in this patient. Amphotericin B, obviously, you had to continue for 14 days after resolution of candidemia. So what's interesting about this is obviously the first time that these were compared, but also a sub-study that was done looking at central venous catheter exchange, which I think comes into play when we're looking at de-escalation. No exchange, 5.6 days versus 2.6 days if the catheters were exchanged. And certainly we know that also from David Andy's paper that was published a few years ago. This was followed up in 2002, Caspo versus Ampho, and there was de-escalation, and again, it was on day 10. Only 25 to 35% of the patients were de-escalated to oral fluconazole. So somewhat helpful, but not very helpful. And then Kohlberg, voriconazole versus amphotericin B. This was a switch either to voriconazole or fluconazole after 3 days. Interestingly enough, the median time to clear the blood culture in this study was actually 2 days. So something else to keep in the back of your mind when you're de-escalating. Riboli and anigilafungin versus fluconazole. This was another one that documented a step down, but only after day 10. So how often was the step down used? Well, 26% of patients that were on anigilafungin stepped down or de-escalated to fluconazole versus 28% of patients that stepped down or de-escalated there as well. The duration of therapy is basically about the same, almost 2 weeks. And then one of the last ones, isavuconazole versus caspofungin. This was again, Part Yan in 2019, where the step down was after day 10 to isavuconazole or voriconazole. This was a little bit higher. The step down occurred in 35% of patients versus 40% of patients. So we have some data that step downs do occur, but they don't occur very often. This is probably one of the highest ones that we have. So I'm going to take you back to 1972, early de-escalation. So for those of you that have been around for a while, Jerry Medoff, Dismukes, Mead, This is a 5-patient study with systemic candidiasis, and they decided instead of the long course of amphotericin B, they decided to do low dose. So recognizing low dose, so they went anywhere between 10 and 335 milligrams in those 5 patients. In those 5 patients, the follow-up was amazing from 2 to 24 months. Clinical cure was achieved, and there were no relapses. So if you could say this is probably one of the first early de-escalations that we had back in 1972. So 95, and then nothing happened until 95, 96, where we had a pediatric population of patients. This was Dunowitz. That was published in 95, 30 pediatric patients with candidemia. And this one actually, after the blood culture cleared, they required, depending on the patient, 7 to 14 days. And when also looked at a high dose versus low dose of amphotericin B, again, conventional amphotericin B, overall cure rate was 60% in both groups. So starting to see a little bit of data actually on what's called low dose amphotericin nowadays. So this was actually an interesting study. This was 99. It was a pilot study. It was a single center study. So Carl Fichtenbaum and his group, not a big study, but for a single center, 76 subjects, 38 were UC, which is an uncomplicated candidemia. And those patients, the definition was less than 1 day of a positive blood culture. They received 200 milligrams of ampho over 5 to 7 days max. 38 subjects had PC or positive cultures for more than 1 day, and they received 500 milligrams, 14 to 20 days. The clinical cure rates, 93% compared to 83%. As you can see there, the overall outcome on day 180, 72% compared to 54. Now, it's not fair because the patient that had positive cultures were sicker, they had sustained candidemia, but it's just to show that in an uncomplicated patient, actually, they do pretty well when they receive a 5 to 7 day course and a short course of amphotericin B going back. 2014, another very small retrospective study. In this study, 37 patients, 70 actually, were stepped down to fluconazole, 1 relapse, 30-day mortality, about 37%. The study that I published actually with Pete and a lot of the MSG, this is a prospective study, multi-center, and it was 250 patients. This was inidulafungin for 5 days, followed by a step down if they met criteria. So this is the first time that we actually use some kind of criteria to initiate the step down or de-escalation, meaning they either had to take oral therapy, afebrile for 24 hours, vital signs were stable or hemodynamic stability, and a negative blood culture. In this study, that was specifically to evaluate step down. How many patients were stepped down? 60%. So better than before, but certainly not a great number. The global response rate was a similar 84% in both, and median time to negative blood culture was about 2 days. Duration of therapy, however, did not change at all. So this is one of the areas that I think has to be evaluated. Bailey and Van der Geest, again, looked at, this is a large prospective study, 647 patients. They looked at early de-escalation by day 5, but unfortunately, out of the 647, only 22% were able to be de-escalated, no change. And this retrospective candidemia invasive candidiasis study, 56 patients initiated therapy with a conic hand, and 32, so at least more than 50% were stepped down. Response rate were pretty much similar, and no differences in day 28 and 90-day mortality rate. So looking at a combination there, so almost 50-50 really. The last two, Jafal, this is a retrospective study looking at 190 patients, and I'm not going to dwell on this one because these patients were either empirically, preemptively, or targeted therapy. De-escalation was only performed on 20% of the patients, so it's kind of hard to look at the data, the results, because of the 3 different groups that these folks had. And then 2021, Moreno Garcia is a retrospective post hoc study of candidemia, looking at 235 patients that met criteria, switched to fluconazole. But again, in this study, out of the 235, 23% stepped down, 77% continued therapy. So you can see basically what we see in a lot of these studies is the step down or de-escalation is not very efficient in a lot of these studies. And again, the response rates are pretty much the same. So where do we go from here? Well, I think where we go from here is perhaps what we've done with Staph aureus bacteremia, maybe dividing it into uncomplicated versus complicated, and using certain criteria to initiate a rapid IV to PO and a short course of therapy or duration. Obviously, we have to make sure that there's no metastatic foci of infection, rapid catheter removal, rapid IV to oral therapy. They have to be, or they should be normotensive, a functional GI tract. If not, obviously, we can't switch to PO. Candidemia, uncomplicated, we'll use the definition from before. And then duration of therapy, 7 days. Is this enough? Well, the single dose echinocandin rezafungin could be useful. This is a one and done phenomenon, similar to what we do with dalbavancin or ritonavir for staph bacteremia. And what about a rapid oral step down with the oral agents available for azole-resistant isolates? So theoretically, abrexafungerp or fosmanogepix could be two that could be de-escalated to, so we can get the patient out in spite of the fact that they may have a Candida glabrata that's azole-resistant or multi-drug resistant. So switch gears a little bit, because this is another one that actually there have been several de-escalation studies. Clinical practice guidelines for Cryptococcus by IDSA was in 2010. And the combination here of ampho or liposomal amphotericin B plus 5-FC for 2 weeks, followed by de-escalation to fluconazole at 400 milligrams per day for 8 weeks, although 2 weeks a lot of times does not cut it in somebody with severe cryptococcal disease. And this is followed by the 200 milligrams, right, of, excuse me, the 200 milligrams of maintenance therapy. The WHO guidelines in 2018 were actually changed, and recognizing that the WHO guidelines are really aimed at lower middle income countries. And that's important because the recommendation here is either the deoxycholate amphotericin B at 1 milligram per kilo per day, plus 5-FC at 100 milligrams for 1 week, and fluconazole daily. Or the other recommendation is a purely oral route of fluconazole at 1,200 milligrams per day, plus flucytosine at 100 milligrams per kilo for 1 week. So these are the current recommendations that we have. We do have some de-escalation trials. This was the ACTA trial that was published in New England Journal of Medicine by Molly et al. And this one actually looked at 3 regimens. An oral regimen, as I just mentioned, of fluconazole 1,200 per day, plus 500 milligrams of flucytosine for 1 week, and deoxycholate amphotericin B at 1 milligram per kilo per day for 1 week. And the third regimen was deoxycholate amphotericin B, 1 milligram per kilo for 2 weeks. Recognizing that all of these patients that are on amphotericin received either fluconazole in combination at 1,200 milligrams per day, or flucytosine 5-FC at 100 milligrams per kilo per day. So a very significant study of 678 patients that were evaluated. The conclusion of that one was the 1 week of amphotericin B plus fluconazole or 5-FC and the 2 weeks of oral fluconazole plus flucytosine were effective as induction therapy for cryptococcal meningitis, certainly, especially in this resource-limited setting. It was also shown, however, that flucytosine was better than fluconazole when you added it to the amphotericin B regimen in this setting. So that's kind of important. One of the ones that I think is really exciting is this one that was published by Jarvis. Again, this was done in resource-limited settings. This was in Tanzania and Botswana. And this phase 2 actually leads to a phase 3 study. But this phase 3 study was non-inferiority trial examining the early fungicidal activity of short course, high dose liposomal ampho. 80 subjects were randomized, so not a huge study, but because they had a control, but liposomal amphotericin B, 10 milligrams per kilo, single dose. So again, once and done. Number 2 was liposomal amphotericin B at 10 mg per kg on day 1, followed by 5 mg per kg on day 3. So 2 doses and you're done. The third one is liposomal 10 and followed by 5 on days 3 and 7. So 3 complete doses. And then finally, the fourth regimen is the 3 milligrams per kilo per day of liposomal amphotericin B. And that was used as control for 14 days. So It is important to note that all of these patients did receive oral fluconazole at 1,200 milligrams per day in combination for the full 14 days. So primary endpoint was mean rate of clearance of the CSF, the cryptococcal infection in that study. And what the study showed was that the single dose 10 milligrams per kilo liposomal amphotericin B was well tolerated and led to non-inferior compared to 14 days of the standard or conventional 3 milligrams per kilo per day in HIV associated. So this is exciting, certainly an exciting study where one dose and essentially you're done and you can follow up with oral therapy. Finally, last but not least, there aren't a lot of other studies. Aspergillus studies are tough to do. The tough to do de-escalation studies. And the reason for that is really a lot of the results, a lot of the outcome depends on the host response. If the host is severely immune suppressed and you cannot reverse the immune suppression, the morbidity, the mortality is essentially 100% or more. Number two is the fact that it's just difficult to make an early diagnosis in many of these patients. So tough to do these, shorter is better, especially because of the host immune system if it doesn't, if it doesn't recover. And in addition to that, when you look at the recommendations, whether it's IDSA or ECMID, many of the recommendations are really a consensus from experts, is what it says. So at this point, IDSA recommends a minimum of 6 to 12 weeks. I'm not going to get into the antifungal drugs. 6 to 12 weeks, largely dependent on the degree, duration of immune suppression, the site of the disease, evidence of disease improvement. And that was a strong recommendation, but a low quality evidence. Why? I'm not really sure. Why the 6 to 12 weeks? Why not 4 weeks versus 6 weeks? Not sure where actually that came from. So this is another area where it's not like I'm new to the area, new to the business since I've been here for 30 years, but I'm not really sure where some of these numbers came from. So in the European guidelines, according to them, consider switching from IV to oral therapy in stable and pharmacokinetically reliable patients. I'm not even sure what a pharmacokinetically reliable patient is, as a matter of fact, but it's an interesting concept. The range of duration here is even broader, anywhere between 3 and more than 50 weeks. Weak evidence to support any of this, and many of the initiation of therapy are using the parenteral route. Why not the oral route in some of these? So again, duration of therapy, 6 to 12 weeks, degree of suppression, site, evidence of improvement as the, and strong recommendation and low quality in this. Other fungal diseases, well, there's certainly not enough to do a step down in Mucor, except the recommendation is to step down from IV to PO whenever feasible. Endemic fungi certainly have not been done, but I do want to point out the duration has been there for quite a while. Blasto, 6 to 12 months. Coccidioides, 3 to 12 months. And I'm throwing these out there because these seem like a long, long time for a lot of these infections. I can see the cocci, histo, 6 to 12 weeks. Some, however, prefer 18 to 24 months. So it's maybe something that we need to consider or look at. So, and something to consider is actually this that was published again by Medoff and Dismukes, and this was in '92, considering using general guidelines for the evaluation of new antifungal drugs for the treatment of fungal infections. Maybe we should relook at this since considering that the recommended dose and duration of therapy on a lot of these fungal infections are primarily based on clinical experience. So go back to '92. So it's been quite a while since this has been looked at to make sure that we follow the same guidelines. We have a long way to go. I think strong support and standardization of all clinical trials include de-escalation or step down, certainly. Studies are difficult to initiate, and once the drugs are approved, they're kind of hard to do. Many of the studies are tough since the response is associated with the underlying disease, immune suppression, and a reminder that oral is generally as good as IV, shorter is as good as longer, and avoid that classic, we have always done it this way, which is what usually gets us into trouble. And with that one, I will end the presentation. I did have a differential diagnosis for extra credit here for, and I think we're going to give away a trip to the Bahamas or something if anybody gets this type of mold, right, Dr. Pappas? So
yeah. Oh,
so
yeah.
I'm giving you 5 seconds to take a look at this and give me a diagnosis. It's actually called Zolaria polymorpha, or dead man's fingers. It's a fungus if you want to eat it and give it a try, but it actually grows on wood, on dead wood. It actually eats the dead wood. So,
Wow.
so we have not seen a human infection with this, but give us some time and I'm sure that some of this will be invading our bodies soon. So
Oh my
with
goodness.
that one, I will thank you very much for allowing me to
Okay.
give the presentation and I'll take questions.
Joe, thank you very much. That's terrific. Thank you so much. Dead man's fingers. All right. I think the elephant in the room is the candidemia that you discussed. And so I think you kind of shared your thoughts on it, but where do you think we're going with this? Are you proposing that the empiric length of treatment that we chose, which is 14 days, should be shortened?
Yes.
And how narrow do you think we can get before we can, or how short do you think we can get with parenteral therapy before switching to something oral? And what are the barriers?
So I think there's two, I think we do have to, okay, so let's talk about the duration first. So the duration, I think what we're trying to avoid with the 14 days, obviously, is extended adverse events, right? Metastatic foci of infection, which we don't see a lot of, specifically in line-related candidemia. You know, we used to think that, you know, the endophthalmitis was very common, or not common, but much more common. We know now that it's not. And, you know, we're having a battle, obviously, with ophthalmologists about that. So I think the duration, if we can do a study, and I realize that the studies have to be done with FDA's approval of 14 days as the classic amount of time. But if we can do an evaluation on day 7 of these patients, you know, and have the other arm continue, I think it's a possibility, you know, of short course therapy. I don't think, and again, an uncomplicated candidemia, not complicated. So if we meet those criteria, and you know darn well from doing many of these studies that you pull the catheter, and a lot of times even the blood culture from that day is negative, right? So what are we doing with these patients? Number 2 is, if the patient's GI tract is functional and they're hemodynamically stable, why not switch to oral? 1 dose of a Candid and then switch to oral if they're hemodynamically stable. So I know a lot of people are switching early, maybe not 1 day, but certainly within 2 or 3 days, a lot of people are now switching to oral. With the new compounds that are available, even if it's Candida glabrata, we're able to switch to oral, right? So they'll be very, very helpful as far as step down. As a matter of fact, they're going to discuss that. Synexus will discuss that later on with the Fury study, right? Which is very, very helpful. So I think we can do it. I think it's time to do it. And I think it's time to shorten the courses and do a rapid switch from IV to PO, which is, you know, what I've been discussing for many, many years, over a decade, I think. So.
Okay. Thank you. There's one other kind of related question that came to you. Joe, this from Jay Aram again. Joe, would a meta-analysis of the invasive aspergillosis, RCTs, allow, what kind of insights would that provide us for treatment duration of invasive aspergillosis? Because talk about an empiric length of treatment. It's extraordinarily broad.
Well, I think a meta-analysis would be useful, similar to what David Andes did with Candida, right? Putting all these studies together, if you could. Certainly the duration is key. I mean, it kind of surprised me when I went through this, how long some of the recommendations are and how much the duration is. So I think it definitely would be very, very useful in that situation to try to obtain some kind of treatment duration, and not only treatment duration, but outcome, which was what was done with the candidemia study as well, right? So.
Yeah. Okay. Joe, thank you. That's terrific. That's terrific. And
thank you
right on time. Really, that was very insightful, and appreciate that.
Thanks for the invitation. Yep.
Our pleasure. Okay. Our last formal speaker today, before we have the breakouts, is Monica Slavin. And Monica is professor medicine. She is in Melbourne, Australia, at the University of Melbourne. She is division director at the University of Melbourne, and involved with Peter MacCallum Cancer Center and the Royal Melbourne Hospital. So Monica's going to talk to us about prophylaxis, preemptive therapy, and empiric therapy. And we'll have a 10-minute discussion, and then after that we will go into our breakout sessions. Monica, thank you for joining us. I don't even want to ask you what time it is there.
Don't ask, Peter. Thank you very much for the invitation and for allowing me to get up at 5 o'clock in the morning. But I'm very thrilled to be here.
You always said you were an early riser. You always said
Okay. So I'm really happy to be here today. And this topic is an interesting one. And to focus my talk, I'm really going to focus on the hematology setting. Although we're talking about antifungal prophylaxis, preemptive and empiric therapy, it's not that clear cut for clinicians. We're dealing with the context of our patients often being on mold active antifungal prophylaxis, the reality that we're seeing more mixed infections, infections with aspergillus and other organisms, the emerging mold infections that we're seeing particularly in patients on antifungal prophylaxis, and the advent of immunotherapies now in our hematology setting, whereby patients, many and frequently may have fever, but not neutropenia. And where does that leave us in trying to decide whether to use empiric antifungal therapy? So it's a murky world, and I'm going to try and discuss some of these issues today. These are my disclosures. So just going back to really set the scene and talk about what we mean by preemptive prophylaxis and empiric. I think we know that with prophylaxis, we're giving these drugs to everybody at risk. We're often using a mold active agent, or if we think the risk is only for Candida, we're using an agent like fluconazole.
Hey, Monica,
but there are caveats.
Monica, let me ask you to get into presentation mode. I think most of us are seeing the kind of the slide sorting mode.
Yeah, I'm so sorry. I am on presentation mode, Peter.
Okay.
So I'm not sure how I can improve.
We're still on the first slide.
No, we're still on the first slide.
Oh, okay. So I'm not sure why it's not progressing. Let me just reshare. I'm so sorry about that.
Yeah, that's okay. No problem.
Yeah.
There's a mechanism.
Maybe you're doing
a swap screens.
Okay, and I'm just going into presentation mode again. Is that better?
It's the same.
Stop share. What I may do is just have to go into that, but enlarge the screen.
Are you working off of 2 screens?
No, one.
Swap screen, top left of a presentation mode.
Did you hear that, Monica? Top left. Swap
Top left.
screen. There's a there's a
I am not seeing that. What if I just enlarge my screen here?
that would be fine if you could. Well, that's too much. That's too much. I mean, we can go from your current one. We just need to, but we're not seeing the advance of slides.
Are we advancing now?
Yeah, you are, but you can just reduce it to its ordinary size, and then I think that'll be fine.
There we go. That's its ordinary size. Is that okay?
I think that's okay.
Yeah, I'm so sorry. I'm not sure what
That's
the
okay.
problem is.
No problem.
But anyway,
No problem.
let's talk about prophylaxis, empiric therapy, and preemptive therapy. So I'm really talking about some of the caveats in these approaches. And so I think really with prophylaxis, one of the key factors we need to know is our risk profile of our patients, because we only want to give this approach to our highest risk patients. And of course, we do have problems when we use this approach in then using serum galactomannan, where we'll have reduced sensitivity. And we are seeing emergence of non-aspergillus molds in this setting as well. Turning to empirical therapy, it's traditionally been indicated for patients with persistent or recurrent fever during febrile neutropenia. And so that is really a limitation in that fever is not sensitive or specific for invasive fungal disease. And particularly in the era of immunotherapies, fever is very common and our patients are not neutropenic often. So we don't really have a sensitive way to determine who needs empiric antifungal therapy in this group of patients. And of course, we still need diagnostics because it's not really feasible in today's world to treat everybody who requires empiric antifungal therapy with a prolonged course of antifungal therapy. Well, turning to preemptive, this is the therapy that's driven by diagnostic tests, traditionally Aspergillus galactomannan and PCR, and use of mold active agent if these tests are positive. Of course, this is more difficult to implement. You really need to identify your high-risk group. You need expertise in interpreting the tests and access to timely test results. So again, caveats around how we might implement this kind of approach. Looking at antifungal usage, of course, with prophylaxis, it's going to be 100% of patients. Empirical therapy studies show us that it's used in 30 to 70% of patients with high-risk hematologic malignancies. And then preemptive therapy, antifungal use is around 20%. So you can see a reduction in antifungal use across these categories. And this paralleled by, of course, cost, toxicity, and drug-drug interactions. But the opposite applies to the logistical challenge. Obviously, it's very much easier to apply prophylaxis to our patients than to apply preemptive therapy, where we need all those prerequisites about expertise and monitoring and turnaround time. So I'm just going to look at a couple of the studies that relate to antifungal therapy and looking at the different strategies. So I want to talk a little bit about prophylaxis studies. And the real point of this slide is not to compare studies. These are very different patient populations. These studies were conducted at very different time frames. And the approaches, some of them have used galactomannan as well as empiric therapy, for example, in the Cornelli prophylaxis study. And some of them have only used empiric therapy without screening in the Marks prophylaxis study. So these studies are not really comparable in their design nor their patient population, but really just to show the findings that we see, the broad trends that we see with these studies. Of course, with prophylaxis, the rates of fungal disease are low and increased numbers of fungal diseases in the empiric therapy approach and the preemptive therapy approach. I would like to talk a little bit about one of the newer studies, which has only been presented in abstract form at the moment by Johan Martens. But this is a very interesting study run by the Europeans. It's a large study, enrolled about 550 patients, where they randomized patients to receive either empiric antifungal therapy, no monitoring, no galactomannan screening, and empiric therapy as guided by traditional means in these patients with AML and stem cell transplant, versus a preemptive approach where galactomannan was used to monitor the patients. And this study was powered on a survival at 42 days after entry into the study. And what's really interesting about this study, I think, is that they do not see a difference in survival between using an empiric antifungal approach or a preemptive antifungal approach. And this is really interesting because I think, many of us have been turning away from an empiric antifungal approach, more in favor of screening and preemptive therapy. But I think we do need more information about this study because I think knowing that galactomannan was available in all of these centres, it's very feasible that once a patient started empiric antifungal therapy, that they were aggressively investigated and received a rapid diagnosis and targeted therapy. I think this is what we need to look at when this study is published, because at the moment we've just seen these abstract and these very interesting figures here. So turning to these preemptive studies, I've summarised here on this slide some of the findings from several preemptive studies that have been carried out over the last several years using galactomannan alone or galactomannan and PCR to screen high-risk haematology patients and to use that as a guide to when to start antifungal therapy and comparing that to a more empiric-based approach. And what these studies have shown us is that it is safe to withhold empiric antifungal therapy. There's about approximately 50% reduction in the use of empiric antifungal therapy if you are using this screening and preemptive approach. But this is offset, of course, by an increased number of treatment courses because you will diagnose more courses of invasive fungal infection with the screening approach. They also tell us that we can diagnose aspergillosis earlier by using this kind of preemptive approach. And as I've mentioned, the Martins study does show that there was a non-inferior survival with galactomannan and CT scan guided approach compared to an empiric approach. And I think this is going to be a very interesting study to look at when we get the full details. Looking at how this preemptive approach performs in terms of cost and in terms of overall cost of the strategy, there have been several meta-analyses, and I'm presenting one here done by Francisco Marti's group, where looking at the cost of the antifungals with the pre-diagnosis of invasive fungal disease and after the invasive fungal disease was diagnosed, as well as incorporating the cost of the testing strategies used in these two approaches, you can see that there was a very similar total overall cost, but that the preemptive therapy came out slightly cheaper in this meta-analysis. But of course, this very much depends on the cost of running the tests in your centre. And if it is more expensive, I think this advantage may well be lost. But several studies and several meta-analyses have found this similar finding in terms of cost. So what are the limitations to these approaches? I think I want to talk a little bit about some of the challenges of implementing these kinds of strategies for antifungal use in the real world. And I guess one of the big ones is breakthrough fungal infections and breakthrough invasive aspergillosis. And a very common scenario is that we are treating a patient with mold-active prophylaxis, and the patient develops a breakthrough. They develop pulmonary infiltrates that's suggestive of a mold infection, and we go on to do further testing, often with a bronchoalveolar lavage or a lung biopsy. But while we're doing that, we'll often start empiric treatment with, for example, a lipid formulation of amphotericin B, wondering whether this breakthrough infection is a mold infection, and if it is a mold infection, is it going to be resistant to an azole? So this is a common clinical issue. And what we also often find is that when we do a BAL, for example, and look at Aspergillus galactomannan, we can detect Aspergillus galactomannan but often do not grow any fungal cultures. And so we're left with the dilemma, is this a mold that this resistant to the mold active prophylaxis that the patient was receiving? Is it truly Aspergillus? And just for reference, I've put a table from a recent review looking at some of the fungi that have been reported to cause positive galactomannan. And you can see that it's a growing list. And so a positive galactomannan may not always indicate invasive aspergillosis. And interestingly, Nocardia has also been added to this list recently. So there are a lot of dilemmas in this clinical scenario. And do we want to leave our patient on 12 weeks or 6 weeks if we follow Dr. Vasquez's advice, or shorter, of liposomal amphotericin B? And this is the big question. We have not enough information really to determine what to do with this patient. So there are some very nice reviews, and this one by Dr. Leonakis, looking at the rates of breakthrough infection with the different prophylaxis regimens, including the azoles and amphotericin B. And what this really concludes is that the breakthroughs that we see are very much related to the prophylaxis that we're using, the local epidemiology, the patient characteristics. So the stage of their cancer, have they been multiply relapsed, and have they had multiple prior therapies, and what prophylaxis they're currently receiving. So there's a lot to think about in these cases of mold active prophylaxis. And I think this slide also gives us some further food for thought, in that this study from Duke looked at patients who developed invasive fungal disease on fungal therapy. And interestingly, the number of Aspergillus infections in the breakthrough infections was much smaller than the non-breakthrough. And the variety of other molds was much more extensive. So we really do need accurate diagnosis for breakthrough infection, because the possibilities are extensive. Looking at diagnostics, if we wanted to really follow down the preemptive approach, this was a survey done by Susan Seo and her group of solid and hematologic stem cell transplant centres in the US. And they had about 71 responses. And you can see that the beta-glucan, galactomannan, BAL-galactomannan, and fungal PCR, they were available on site in less than 50% of centres. So this white here means they're available on site. The grey means they're a send-out test, and the dotted line means they're not available. So you can see this was published in 2017, so things may have changed in that time, but I think this is a very real-world demonstration of the fact that we have these diagnostic tests, but they're not immediately available to us, even in transplant centres in the developed world. But when we look at Asia, the situation is fairly similar, so it's a global problem. The Asia Fungal Working Group recently did a survey looking at what was available in these countries in Asia. And you can see that while most had access to histopathology and pathology, galactomannan was only available to 60%. And overall, 74% of people in this expert fungal working group used empirical approach and empirical antifungal therapy. Interestingly, only 30% had antifungal stewardship programs in their hospital, and 80% couldn't use their preferred antifungals due to cost. So this really shows the global kind of barriers to adopting probably what we would consider to be best practice in diagnosis, and therefore the limitations of the diagnostic-driven approach in the real world. So in the last few minutes, I want to think about how we can improve antifungal use. And we've talked about in this session the new antifungal drugs, which are very exciting. And we've talked about implementing antifungal stewardship and the way we might do that. But really, these two factors are really going to be limited by what we have available to us in clinical practice. And that means the diagnostics we have, the accurate risk prediction tools we have to look at who is at highest risk and who should be receiving these agents. And of course, although I'm not really going to touch on it in this talk, immune reconstitution, which is really the elephant in the room. We've heard it mentioned already today as one of the key factors in improving our patients' outcome. So talking about the gaps and how we might address them going forwards, I think risk assessment is really critical. It underpins who we give antifungal prophylaxis to, who we consider for empiric antifungal therapy, and also in which patient groups we might look at preemptive therapy. So unless we've got good risk assessment tools, we're really applying these strategies in a bit of a vacuum. So I think one of the things that we have heard in recent times is the focus on potentially identifying genetic risk factors to add to our clinical risk factors. There's a lot of work going on about the microbiome and colonization and the microbiome and how that might predict invasive fungal disease. And of course, immune profiling may also play a role in identifying our highest risk patients. So I just wanted to put up this very nice study, which shows some elegant work looking at the CARD9 gene defects and how that impacts on invasive candidiasis. And the bottom line is in these patients and these mice who are CARD9 deficient, they did not form immunoglobulins in response to the gut microbiota, in response to the candida in the gut. And when these patients then got an invasive candida infection, they got a disseminated and serious infection in comparison to those who were CARD9 sufficient. So again, this could be a very useful marker going forward in trying to identify our highest risk group for candidiasis. And again, some other work from Memorial Sloan Kettering looking at how they could predict the patients who were about to develop candidemia by looking at the microbiome and looking at the fungal candida concentrations in the gut and in the stool, and showing this constant increase in concentration until the patient here developed candidemia in the grey bar. So this is again a potential marker for patients at risk for candidiasis, but it's also a potential intervention in the future for predicting and preventing candidiasis. I wanted to talk in the last couple of minutes about diagnostics because we obviously rely on diagnostics for our direction of antifungal therapy, not only in the preemptive approach, but even if we're using empiric therapy, we still need good diagnostics to know whether to continue it, how long to continue it for, and what we're actually treating. So improving diagnostics is really, I think, going to be a key factor for us in better using the antifungal tools that we have. So I've just put up a little summary here of some of the diagnostic tests. And those in bold are those where there are recent publications in the last several years. The lateral flow device obviously could increase accessibility to galactomannan and to Aspergillus antigens beyond tertiary centres that have laboratories equipped to do this in a timely fashion, as could the urine Aspergillus antigen. And I'm not going to talk about any of these in depth, but I think I just point them out to highlight that there is work going on in trying to develop the tools that we need to improve our use of antifungals. Of course, the Mucorales PCR assays are very interesting, but have only been used in a small number of centres and not widely evaluated. This would be a great tool for these breakthrough infections where we're not sure what we're treating and whether you're treating Aspergillus or another mold. And of course, the work that's beginning on metagenomic sequencing of blood and body fluids, such as bronchoalveolar lavage fluids, to rapidly identify fungal genes in the tissues and fluids, is also, I think, quite promising. And in the imaging arena, the PET scan is something that we're using more frequently because of its metabolic activity. So that when you see a pulmonary infiltrate or a fungal or a nodule, you're able to look at the activity and the metabolic activity, which is often helpful in distinguishing scarring in the lung, fibrosis, and active fungal infection. It can be used to follow infection on treatment. So these, I think, are all things that we are coming along in the future that will help us determine how to use our antifungals better. So in conclusion, I think that we can say prophylaxis, empiric therapy, and preemptive therapy, the jury's out. I don't think one is superior to the other, because the tools we've got in employing these approaches are quite blunt. And as we can see from the studies that I've shown you, it's often not an exclusive approach. There's often an azole prophylaxis plus an empiric approach, a fluconazole prophylaxis plus a preemptive approach. Early reports do suggest that there's no mortality difference between the empiric and preemptive therapy, but this will probably depend on also the diagnosis and the type of patients. And I think we need more detail from this very interesting European study. An empiric approach may be less useful in patients receiving immunotherapy than in the classic febrile neutropenia population. Less antifungal use has been shown with the diagnostic-driven or preemptive approach, and lower cost as well. But again, this very much depends on your local access and your local availability. So future research on risk prediction for fungal disease, microbiome modification may help prevent infection, and enhancement of diagnostics will improve availability and help identify breakthrough infections with non-Aspergillus molds and better guide therapy. So I hope I've sort of summarised the field somewhat and some of the dilemmas that the clinician faces in knowing which is the best pathway to follow here in the antifungal arena. Thank you.
Thank you, Monica, for an outstanding review. I think maybe what you didn't highlight is just the differences in availability and usage of some of the diagnostics and what drives empiric and preemptive antifungal therapy in the U.S. compared to other regions. So, for instance, I mean, I think we are much more empirically driven, much less reliant on diagnostic tests. I don't quite understand why that is. I don't know if you have any insights into that or not, but I know the Europeans and probably you in Australia are more dependent on the fungal diagnostics than we are. Maybe they are more reliable, or you've just been brought up in a manner to trust these. You have any explanation for that?
I think there's a number of points there, Peter. I think, yes, the fungal diagnostics are very much relied on in our setting in Australia, but that comes with a caveat, and there really needs to be the expertise to interpret them. As we've heard from the excellent talk by Dr. Dodds-Ashley, if you can't use these tests in an area where you don't have an expert actually looking at the results, because they're not a black and white test, and you need to take into account, is it a true positive? Is it a false positive? Do I have a repeat positive? Do I have other supporting evidence with CT and PCR? So it's actually quite a process, as you say, using these tests. And that doesn't even account for whether the laboratory is set up to do a quick turnaround time. We are fortunate now in our laboratories in many of the tertiary centres can run these tests twice a week, the galactomannan and the PCR, but you really need that in order to take on this approach. I would say that empiric therapy is a very blunt tool, and my concerns about using it are that you actually don't know what you're treating and you don't know how long to treat it for, and it becomes with successive cycles of chemotherapy, it becomes, you know, standard therapy to continue it, so for very long periods sometimes. So I think from an intellectual perspective, it is better to know what you're treating, and you will reduce your use of empiric therapy. All the studies have shown that you will reduce your use of empiric therapy, but the flip side is you will diagnose more infections and be treating more probable fungal infections. So I'm not sure why the culture is different in the US. It's an interesting question.
I've never quite understood it, but it is the world in which we live. I'm going to thank you again. That was outstanding, and thanks for getting up early to join us. We reached the part of the program where we have breakout sessions, and I'm going to ask Debbie to kind of instruct us through this, because we're going to have several choices. Can you get on, Debbie?
Yeah, I'm here. Okay. So everyone, I have just opened the breakout rooms. If you can, look to the bottom of your screen and select that breakout room option, and it should open up to your options of rooms to join. From there, you can choose which room you'd like to attend. And if you need any help, just let us know. I'll be here in this room to help out.
And what about joining? Can one go to different rooms?
Yes. Once you are done in a room, you can come right back to this main room and then join a different room.
Okay. All right. Okay. I would encourage you each to choose an area of interest and join those rooms. And if you want to move to another room, then do that as well. Okay. All right. Well, we're at the end of our time. I can't express how much I enjoyed these presentations today. I thought everybody did just a fantastic job. So short, succinct presentations, lots of good questions. Of course, I only attended one of the discussion groups, but it sounds like at least some of them went on beyond the allotted 30 minutes. So I want to thank everybody for participating. I thought this was really useful. Before closing, I'm going to just alert you to the fact that you will be getting, if you participated today, you will be getting a message regarding feedback. And so just be honest in your feedback. What can we do differently? How can we make this better? Anything that you think will be helpful to us to kind of perfect these webinars. Again, as I indicated, and as we did in November, this webinar was created in part to kind of decompress the main program for the biennial meeting, similar to the COVID fungus webinar that occurred in November. So we may be looking at one more, and then we will be hearing shortly. All of you will be hearing shortly as to when the date for the biennial meeting will be. It'll be sometime between August and November. And actually, after these multiple delays, we'll be back on target with our even years, alternating with Tim. So two things to be looking for. One, the announcement for the Tim meeting. I mean, for the biennial MSG meeting in the fall. And then secondly, and most urgently, please get those assessments back to us as quickly as possible so that we can make these webinars better. Thanks again for joining.
Peter, can I interrupt just a second?
Please interrupt.
I agree with you. These talks were absolutely outstanding. can you say how these things are captured and are they put on Dr. Fungus or someplace that people could go and get these things when they weren't here? Not lose these things, because they were incredible.
I thought they were very, very well done. Can I get somebody from the main?
Yep. This is Debbie. We have recorded all of the sessions, including the breakouts. If handouts were provided, we will be distributing the handouts as well. The videos will be compiled as one video recording and then also as individual recordings. All of the recordings will be placed on the MSG ERC YouTube channel and available for future viewing. Does that answer the question or anything else?
No, perfectly. Thanks for doing that.
Yeah. Also, if you go to covidandfungus.org, Terra Nova has snagged them and put them there. We had one of the webinars that was dealing with COVID and fungus. And so we put all of those there so that you don't have to go to a YouTube channel, but they're right there.
Yep.
Pete, this is Debbie. Also, I didn't know if we wanted to ask just for a very brief, maybe summary or a comment on the discussion in each one of the breakout sessions. We have about 10 minutes of time left. I don't know that that's enough, but I'll leave that up to you.
I'm perfectly happy to do that. Let me, I will summarize. Our group talked about ATI 2301, new compound. Think about it as a compound that is parenteral and is compound that really inhibits mitochondrial function. It's broadly active against fungi, but it's being targeted towards primarily multi-drug resistant Candida and crypto. John Perfect has some really nice, really compelling data for activity in his crypto rabbit model, I believe. And so those two areas are going to be areas of potential areas of development. It also comes out in the urine in reasonable quantities, and so it can be considered as a compound that might be looked at as a compound to treat multi-drug resistant Candida in the urine or in the kidney. Those are my summary comments. John, which group did you do?
Okay, Pete. So we did with Jay Abrams, Pfizer on the. We didn't talk about phosphomatogipix. That was discussed by Andre.
What you talking about? We're talking about Duke basketball?
No, we just asked questions. So Jay asked me 4 questions, and I asked Jay 4 questions. We almost got through my final questions to Jay, not quite all the way, but those questions should be. I guess Debbie will capture those things, and it will be in that discussion of what we were trying to do. So Jay challenged me to some things, I challenged him to some things, and that's where we were at. We were doing a lot of philosophizing and opinions.
Okay, all right. Luis, what group did you lead?
I had the pleasure of leading the F2G group with Emma. Emma did a really nice comprehensive review of F2G, the molecule, the development program, and we asked a few questions about PKs, PDs, clinical trial design for the upcoming Aspergillus trial. And one of her main questions was, for us, was what obstacles do we see in getting the new antifungals on formulary in the different hospitals? And in general, what obstacles are we facing going forward with the use of these new compounds? Very, very, very nice session. Thank you, Emma.
Joe, did you chair a session?
Yes, I did. I breaks the fun group session.
Okay.
So David, yeah.
Can you say that? Can you say that 5 times?
I breaks the fun group. I breaks the fun. It's kind of like isavuconazonium.
There you go.
So actually, David presented, he actually presented some data, first going over what exactly is ibrexafungerp, because it is a novel, glucan synthase inhibitor. He went through the oral bioavailability because of the study and the drug that's approved now for vaginal candidiasis, the activity, including the fact that now there is mucor activity, which is great to hear, certainly another drug that's necessary for mucor. the favorable profile, and then he went through the different studies that are being currently done or planned, including what's called the Mario study. And the Mario study, for those of you that have not heard, is the phase 3 randomized oral step down for invasive candidiasis or candidemia, which should be initiating very, very shortly. So that's good to hear. And then the refractory, a little bit on the Fury or the refractory study, we discussed the excretion in the urine because it's always a problem. I think that we have is Candida glabrata. When you see glabrata or Krusei in a lower urinary tract infection and its efficacy. So apparently there's effect in the kidney for pyelo, and there appears to be some urinary levels, although low, they are above the MICs. And I think that took us through the entire timeline. So,
Okay. All right. There were 2 other breakout groups. Who led those, Debbie?
Dr. Lionakis and Dr. Speck.
Michalis, can you give us a brief report, Michalis?
Yes, absolutely. Hi, Pete. Hi, everybody.
Hey, how are you doing?
Good. Considering everything, I think we're managing.
Good.
So Terry presented data on cochleated amphotericin B. He started by an overview of the lipid nanocrystal technology, and then went into what the group thought very exciting early data on the ENACT trial, which is step down initially and early treatment, induction, and consolidation for cryptococcal meningitis in patients with HIV, showing efficacy, early mortality, and as well EFA, which was the primary endpoint of this, while being very, very safe. So there were questions that related to what the next steps are, which appear to be a direct comparison between conventional amphotericin with cochleated with 5FC from start to finish, meaning from induction. And then there were questions with regards to the right dose, which appears to be locked with this new study. I can finish by saying that this drug appears to not have the nephrotoxicity that we're all familiar with amphotericin. It doesn't get excreted in the urine. And that's what their experience with ENACT is. And the few patients as a proof of concept we treated here at NIH with chronic mucocutaneous candidiasis with cochleated amphotericin B over 4 years, each did not develop any renal toxicity even after 2 or 3 or 4 years of being on daily amphotericin. So it was an exciting session. Thanks for having
that's a remarkable observation in those patients with refractory mucocutaneous disease. Who led our last group?
That would be me. So there we talked about resifungin, and we mostly talked about the two phase 3 trials. The enrolling prophylaxis trial, where we'd be, it's a one drug to prophylax for all the fungus, including PCP, as well as the top line results of the phase 3 and the combined phase 3, phase 2 trial, which is showing better early outcomes in the resifungin group in terms of culture clearance and mycological eradication, but no signal at this point on mortality at the 30-day mark. And we discussed some of the side effects that that are more prevalent with other echinocandins, but not so much with this particular compound, as well as the studies that were done preclinically that didn't really note FKS mutations and a rise of echinocandin resistance in the long tail end of resifungin.
Good. Well, this was the first time we have done this, and I thought it would go well. It sounds like it went pretty well. Sounds like the discussion was robust, and 30 minutes may not have been enough for everybody, but I'm glad we did it. Okay, so we are at the 4 o'clock point now, and so we will let everyone go. Again, I want to thank everybody for their participation. Thank MSG ERC headquarters, Debbie Zapponi and her group, and Carolyn Jones, and all of the speakers and organizers and those who helped to precept these sessions, and everyone who participated today. Thank you so much. Please get your assessments back to us, and y'all have a great day, great weekend. We'll be reaching out very soon with a firm date on the biennial meeting in the fall. Okay. Thank you. Debbie, you have anything more to offer?
No, I think it was a great session. Thanks, everyone.
All right. Thank you, Debbie. Thank you, everyone. Have a great day.